So as I was saying, MLCB has been around since 2004, and until 2017, it was basically an official NeurIPS workshop. In 2019, we basically became an independent conference. We were mostly co-located with NeurIPS. This year, due to COVID, of course, we have to be virtual, and so that's what we did. We've had an amazing turnout, so thanks to everybody who submitted abstracts and who registered for the conference. We have at least 1,000 people registered for this meeting. So that's been an incredible turnout. In fact, we can't host everybody on the Zoom webinar, and so we actually have a live YouTube live stream going on at the same time. So I want to thank firstly all the co-organizers, David Knowles, Suin Lee, Sarah Mustafavi, Gerald Kwan, and James Zhou. And just to give you a few statistics, as I mentioned, we have about 1,131 participants registered for the meeting. We had a pretty record turnout of 91 submissions for papers, from which we had to somehow, they were all peer reviewed, and we eventually selected the top 14 for oral presentations. And we have 9 spotlight talks and 39 posters, which will happen at the GatherTown site, which you can get to from right here. Okay, so just a few other discussion points for the meeting today. Please post your questions via the Q&A section in Zoom. Please don't put questions in chat. It's much easier to handle the Q&A through questions through the Q&A section of Zoom. And the moderators, we will read out your questions. So you won't get a chance to actually ask them with your voice. Type them out while the talk is going on. And at the end of the talk, we will curate these questions and try to get them answered by the speakers. Have I missed anything, James or David?
Sounds good.
Okay, excellent. So let's jump right in. We have a fantastic schedule for today. Super excited about 3 amazing keynotes. And also on Tuesday, we're going to have a panel discussion, which is also going to be super interesting. So we'll start off with our first keynote speaker. I'm going to stop sharing my screen. So it's my great pleasure to introduce Aviv Regev. Aviv needs obviously no introduction. She is currently a computational biologist and executive vice president of Genentech Research and Early Development. But most of you probably know her as a professor at the Broad Institute of MIT and Harvard, where she was just until a few months ago, and at the Department of Biology. Aviv has been at the forefront of genomics and computational biology for forever. She has obviously done amazing work in the field of computational models for gene regulation in the early days, and more recently has been really a leader in pioneering efforts for single-cell genomics, both experimental and computational techniques. She has been awarded a ton of really great awards for her research, including the Overton Prize, the ISCB Innovator Award, the NIH Director's Pioneer Award, the Burroughs Wellcome Fund Career Award, and more recently, just in 2019, she was elected as a member of the National Academy of Sciences. So we are super, super excited to have Aviv with us today, and she's going to present her keynote talk on design for inference for tissue biology. Thanks so much, Aviv.
Thank you. Let me share my screen. Just give me 1 second. Where is it? Can you see it?
Yes, perfect.
And now I put it in full screen mode. Can you still see it?
Yes, it looks perfect.
And I apologize, there's some noise in the background, but it will go away very soon. Okay, so it's a great pleasure to be here today. And also I want to thank the organizers for inviting me. It's a lot easier to go to meetings these days than in the past, since you don't go actually anywhere. But I did do the new thing that I do when I when I go to these meetings is that I try to quickly look at the list of participants, which is the equivalent of who you run into in the coffee break, I guess, these days. And so a lot of familiar names of, old friends and also new ones. So thank you for joining us. And so I'll start with my disclosures. I'm a co-founder and equity holder in Celsius Therapeutics and Immunitas. And until July 31st, I was an SAB member with either cash or equity compensation in Thermo Fisher, Neogene, and Syros. And since August 1st, I'm an employee of Genentech and Roche, and my lab is moving there. But all the work that I'm showing here, naturally, given the amount of time that passed, which is very short, still comes from my lab at MIT, the Broad, and HMI. And so biology is the integration of multiple levels of organization, and the fundamental unit of life is, of course, the cell. But in multicellular organism, the way that cells function really depends on the balance of the interactions between them. So we have, they form these direct interactions. These are assisted by the tissue structures that they form, and then they assemble the organs that eventually account for the organism's physiology. Now, tissues are complex, they're structured, and they're dynamic cellular ecosystems. And in this way, they actually maintain this remarkable feat known as homeostasis, which, of course, when it breaks in different ways, we end up with disease. Now, to accomplish homeostasis, cells need to somehow join in their action. And they do this through a process of division of labor that really characterizes multicellular organisms between the cells that are kind of the core cell of the tissue, sometimes known as the primary cell or as the parenchymal cell. So, for example, alveolar epithelial cells in the lung or neurons in the brain, together with accessory cells, which are basically all these other cells that are in the tissue, the stroma, the neuron, the stroma, the glia, the immune cells, and so on, which help those cells. So in the case of, say, an alveolar epithelial cell, this would be the alveolar macrophage and many other alveolar cells. Now, dissecting tissue function really requires us to understand, to do molecular and cellular experiments that would give us mechanism and genetics in order to obtain causality. And so, as is often the case in biology, structure tends to go together with function in ways that are interesting to us as biologists. And so the way that cells are organized physically in the tissue, in these spatial and histological patterns, can really reflect their joint functionality. And so I will call these things histological modules. And to dissect tissue structure, we also need to know certain things. We need to know the molecules and the cells and how they're organized histologically, which is another thing that we typically measure experimentally. Now, what's difficult about these kinds of relationships, say from genes to cells and from cells to tissue, is that they're not linear, either structurally or functionally. And so, at least until recently, if we wanted to relate molecules to histology, we needed to measure these molecules directly in situ. And for the functional question, if we wanted to genetically, if we needed to, genetically manipulate any combinations of genes and measure the outcome, because we didn't know how to predict those. And so today I'm going to talk about some of the ways in which I think algorithms can help in thinking about both the practical and the conceptual challenges of tissue biology. I'm not going to have an answer for all of the problems that I opened up, but I'm going to highlight several of the ways in which our own lab and other labs, I believe, are trying to tackle some of these challenges. And I'm going to cover three kinds of examples. The first is how to define new experimental modalities based on algorithmic ideas. The second is how computational methods can help address the limitations of current experimental modalities and also enhance and integrate data so that we have a better understanding of how the different pieces come together. And the third, and I think the most interesting one, but also the earliest one, is how we can help use these approaches in order to make new biological discoveries and give us a conceptual framework with which to study tissues. So I'm going to start with new experimental modalities and remind you that today, if we want to know where cells are, we typically have to measure them in situ. And there's a range of approaches for how to do this, both at the RNA and at the protein level, but they all have trade-offs. And so one of the key trade-offs is between resolution and genomic scale. Many of the high resolution methods rely on these predefined sets of probes or antibodies, but their number of probes and antibodies, or what I would call the number of measurement channels, is usually limited because of instrumentation limitations, most of all. And so several years ago, Brian Cleary, when he was a graduate student in the lab, suggested that maybe we shouldn't measure each gene individually and could still recover the information that we need. And so, for example, if instead of measuring each gene separately in each sample, we could rely on approaches from compressed sensing to collect compressed measurements of gene expression and use the decompression algorithm to give us individual gene levels with some, theoretical guarantees that that should actually work. And so these ideas are used in other domains like signal processing. We can acquire, for example, compressed images. And this works well because images are structured and not random. And gene expression is also very structured. This is not a new insight. It's been around since the very early days of functional genomics, and actually since well before then from our knowledge of regulons. And so sets of genes can be co-regulated, going up and down together in sets of samples. And these modules are used in different combinations by cells to give us eventually the cellular state that we observe. And so could we compress expression data as well? The reason that we wanted to go after this is that it would be very helpful because as I told you, there's many measurement methods that have a limited number of channels. So if each channel is used for just one gene or one protein, they can only measure maybe dozens of RNAs of proteins. But what if we could use the same 100 channels and get the level of 10,000 RNA or proteins instead? So how might we do this? For this, we introduced the idea of not measuring individual genes, but measuring the abundance of a smaller number of what I would call composite genes, where the composite gene is basically a linear combination of different gene abundances. And so theory suggests that we could choose the coefficients in this linear combination randomly. And specifically, we showed that they can even be binary. And that's important because for many activities, it doesn't matter exactly how you set the coefficients. But for experimental biology, for a lab implementation, binary coefficients are the easiest thing to do. They're just a summing up of whatever is your detection method for the individual molecule. And so first, let's just imagine that we somehow can collect this kind of data. How should it apply in practice? Are my slides still advancing? Because I got a weird message on the screen. Do you see now a white slide and it just says compression for transcription? Great, thanks.
There's a little bit of a shuffling sound occasionally, but otherwise it's fine.
Okay, I have no idea what the source of the shuffling sound is. Before I knew it, there was some work outside, but maybe, okay, I'm trying to reposition my laptop. Hopefully that would help.
Yeah, it's fine. It's good.
Okay, so one option is, so imagine that we could make such measurements. How would we get individual gene levels from them? So one option is to use a little bit of training. So the idea is that we'll take, say, 5% of the full data that we would want to measure, and we would measure it fully, full gene expression profiles. And we would use this data in order to find structure by fitting some model where the expression of the genes across the samples is going to be explained by a dictionary of gene modules and by the activity levels of the modules in the samples. And you can use different approaches to get something like this. You could use SVD, you could use sparse NMF. We tried both of them, and we also developed our own algorithm that we called sparse module activity factorization, or SMAF, because it required the model to be sparse in both the module dictionary and the module activities, and that is beneficial for decompression later on. Then imagine that we take the remaining 95% of the samples and we only obtain compressed measurements for them, which in this case we actually did with simulations first, and we do this with random weights. And so we don't know anything about which compositions to use, we just use random compositions. And then we use the module dictionary that we learned in the first step and the compressed measurements in order to predict the levels of individual genes or to decompress the data. And of course, if we did it in simulation, we can then compare the original and the decompressed data and see how well we did. I also want to point out that in principle, there is theory to suggest that we could also decompress compressed measurements without any training at all. That's called blind compressed sensing. And in biology, it would mean that we would infer the level of each gene, even if we've never seen a full profile, only compressed ones. So BCS, which is used in signal processing, requires you to make some assumptions on how the data basically is structured. And the kinds of assumptions that people have made for other problems in signal processing are not good biological assumptions. So instead, we made the assumption that samples that cluster together when in their compressed measurements likely activate the same kinds of modules, which is a sensible assumption given what we know about biology and gene regulation. And so Brian devised an algorithm that he called BCS SMAF that first clusters the compressed samples And then we search for small dictionaries of modules per cluster. And then we concatenate them to generate the module dictionary. And this is the starting point for an iterative optimization on the modules and their activities. And so now we would ask whether all of these ideas could possibly work for biological data. And so a couple of years ago, we showed that in theory, this should actually work. And we did it by simulating on either bulk or single cell RNA-seq data, taking full profiles, compressing them in simulation, applying the methods to them, getting decompressed results, and seeing what we do. And actually, the first test that we did was without any decompression whatsoever. We just took the compressed profiles generated on GTEx data, clustered them, and saw that we got the same clustering as we would get from full profiles. This is actually a theoretically expected result. We shouldn't be that surprised. The second was that we can correctly recover abundances after decompression. This example is from blind BCS, and we perform even better if we have the 5% of training data. And then finally, we also did it in the context of perturb-seq data because we thought that phenotyping perturbations in compressed regimes is particularly beneficial for screens where we have a very large number of experiments. And so more recently, we wanted to try and move this to practice and to see if we can do this in the lab. And so we chose a great lab assay called HCR-FISH that can measure only a small number of individual RNAs, but at a very high resolution. And so the way that it works is that you do a series of single molecule FISH experiments in one section. And in each cycle, you measure 3 different channels, which typically would mean 3 different genes. And then you can do it through 4 or 5 rounds to get to the full number of genes that you can measure in this assay. And so in order to modify this to a compressed setting, we used a 2-step design. In the first step, we did use training data from single-cell RNA-seq, not from a spatial measurement technique. That allowed us to find the gene modules and define gene compositions. And we reasoned that, doing single-cell RNA-seq today on a piece of tissue is actually not that difficult. Why not use it if you're allowed to? Then we're going to use our channels to perform composite imaging of these combinations. We will recover the module activities, and then we will decompress the image to the level of individual genes, because in imaging, it is difficult for us to get to large numbers of genes. And so to do the compressed measurements in what we call composite in situ imaging, or SISI, we measure each color in each cycle. In each color in each cycle, we're going to measure an aggregate of probes from multiple transcripts together, simply by using probes with a single color that target multiple genes. And you can see that the genes repeat themselves in these different composites or combinations. And so we did this together with Fei Chen's lab, and we measured 9 composite images of 37 genes in the mouse cortex. And if you know the mouse cortex, this is not like any individual gene would look like, because these are the composite measurements, and you see very little pattern apparent to the eye. Okay, I have to actually, now my computer is trying to restart itself. So give me a second before that actually happens, which would be a very bad idea. Okay, my apologies for all these. Can you still see my slides? Thank you.
Looks good.
And can you see the next slide now with an encoder and a decoder? Great. Okay. I don't know, the Zoom is really conspiring against me this morning. Okay, so now we need to decompress these compressed images to get real images of real individual genes. And we had actually 2 approaches for this. The first one that is not on this slide is that we first segment the cells, and then we decompress the expression per cell. So that's more similar to what I showed you before, and really just relies on expression levels. And we do this by estimating the gene module activities and the individual gene abundances. But what is more interesting from my perspective was that we also developed this segmentation-free decompression, which is just based on a convolutional autoencoder that would infer individual gene abundances at each pixel in the image. So here we do the decompression from 10 to 37 channels, so 10 composite to 37 genes in the encoded latent space, and then we decode those images with a decoder. And so that's a different approach, and it has some benefits. The first one is that you don't have to segment upfront, which is a painful problem. You don't have to spot detect, which has benefits in the imaging, which I'll get to later. And the third piece is that you can actually benefit from all sorts of patterns that exist in the image itself that reflect on the dependencies between genes. Okay, And what happens when we actually use this? And so when we decompress, we get these very precise and accurate patterns, which I'm showing here for 3 genes, SST, VLC17A7, and VIP. You should realize that none of these 3 images was actually acquired as is under the microscope. They were only parts of different composites. And so the pattern, though, fits really well. First of all, when we compare it to the Allen Brain Atlas, which you're seeing on the left, but also when we individually measured these genes on the same actual sections at the end of the process. Remember that this is a cyclical process, so you can leave the very last step not used for composites, but used for individual genes, which we do repeatedly through multiple cycles. And as a result of that, we have a ground truth to compare to. And so the red signal is directly measured, the green signal is recovered from decompression, and the yellow pixels are the ones that overlap. And you can see that the overlap is really nice down to the pixel level. And then finally, we extended this to a much larger data set of 12 coronal sections, 180 millimeters squared, almost 500,000 cells, 37 genes in 11 compositions. And we didn't just use genes that were good, cell type markers, but also genes that were characteristics of cell states. And we got a good reconstruction after the decompression, both of the cell type and of the cell state genes, which were mostly immediate early genes. And we also recovered cell cluster labels and spatial patterns. And because we multiplex the genes, we don't need to do spot detection. And as a result of that, we can use just 20x imaging. So overall, we gain 100 to 300x in efficiency. And so this idea can, in principle, be applied in a very similar way to proteins too. These are some results from simulations, which we did with data in collaboration with Ron Germain, but we have not yet optimized this experimentally. And so to summarize this first part, I showed you that because gene expression is really structured, we should be able to use compressed sensing by measuring random composite measurements and then decompressing them. This requires, though, shared regulation. And so if you're looking at things like, say, assist EQTL, where a gene is very specially and separately regulated, this is not going to work. But very nicely for us, most of gene regulation is actually shared, so it seems to work quite well in practice. We can decompress from 200x if we use a 5% training set, and we can even apply decompression to compressed measurements blindly, and so with no training at all. And we applied this experimentally, and we showed, in this way, we showed compressed imaging for RNA, and we have this nice segmentation-free decompression method, but also a more segmentation-based method. And they have kind of pros and cons, each of them. And so in addition to getting 4-fold more multiplexing, and that will grow the more measurement channels you have, we can also work at much lower spatial resolution. So overall, we get this 100-fold to 300-fold gain, which is very important in experiments that are costly and timely and take a lot of time. And so this point that algorithms should lead to new experimental methods and design is also behind another method, DNA microscopy, that I'm only going to mention very briefly. And so this was pioneered by Joshua Weinstein, who now has a lab at the University of Chicago, but used to be a postdoc with Fang Zhang and myself. And today, if you actually want to measure molecules in tissue, you have to choose between one of 2 imaging modalities. The first is optics, going back to the 1600s. And the other is to physically capture things from a known location that you track along, and later on you can reconstruct. MIBI works in this way, for example. And so Josh came up with a third imaging modality, which is both optics-free and registration-free, and is really just based on a standalone biochemical reaction. So really briefly, he codes every transcript with the UMI in situ, and then he encodes the proximity between transcripts by what he calls a unique event identifier, or a UEI. So these are unique barcodes that would form when a pair of diffusing copies of 2 distinct molecules are going to meet each other. You let this diffusion happen on the tissue or in the cell culture in situ. And then after sequencing, you can decode an image from these UEI frequencies that are associated with each pair of UMIs, because you're going to rely on the fact that given diffusion in a structured environment of cells, the more proximal the sources were, the higher the probability that there would be more UEIs that would form between their, diffusing fronts of their copies. And so Josh demonstrated DNA microscopy that it works well for small and large gene signatures, and even transcriptome wide. And so algorithms can really modify existing modalities, or they can suggest entirely new kinds of experiments. And DNA microscopy is an example of this, optics and registration-free method, just a standalone chemical reaction. The resolution depends on the sequencing depth. You can think of the read as 0 photon. It actually works out in the math that way. And we think that it would be really important for these mutation-rich types of tissues like tumors and immune systems. Okay, so this was the first part. It's about how you can think beyond what we can do experimentally by taking an algorithmic approach to experiments. However, in many cases, our computational methods really focus on taking data as it is from other methods and trying to address their limitations or enhance them in different ways. And so I'm going to focus on the same trade-off between the spatial resolution and the genomic scale. And one particularly helpful approach has been to use algorithms to augment spatial measurements by using additional data sources, and in particular by using single-cell profiles. And so quite a number of years ago now, Rahul Satija, when he was a postdoc in my lab, he's now, a tenured professor in NYU, together with Jeff Farrell, when he was a postdoc in Alex Shearer's lab, he now has his own lab at the NIH. This was a while back. They wanted to find a way to relate single-cell profiles to space, in this case, in the context of fish embryos, where they had spatial patterns that were measured in situ. These are all in situ hybrids, ISHs, one gene at a time. And they had the single cell profiles. And so their idea was really clean and simple, like all good ideas. You start with an in situ pattern for a bunch of landmark genes, X, Y, and Z. Together, they would form our reference map. Now we would take a cell. It expresses, say, the red and the blue gene, but not the yellow. They would predict it comes from here. It expresses the yellow and the blue, but not the red. They would predict it comes from here. And you need to do some extra things, address noise and dropouts and matching the different types. The nature of the data is different, so the distributions are different. But at the end of the day, they developed an algorithm for that called Syrah, then got, the name got reused much more broadly. And it handled the noise in our data and did the mapping probabilistically. And one of the nice utilities of this approach was that once we mapped the cells, we can use the mapping to infer spatial expression patterns of genes that were not actually in the in situ data. So if these are the mapped cells and there's some gene X that was expressed at some level at each cell, we can now infer an in situ pattern for the gene in each bin by weighing its expression in each mapped cell with the probability of mapping that cell to the bin. And this approach has since been developed substantially by different groups in different ways. And I should point out that when we published it, John Marioni published a very similarly conceptual method, slightly different technically. We did it back to back. And so this is kind of the beginning of the type of integration that one would need if you want to build an atlas. It's not just to improve the ability of the lab method, but we really want to relate across these different scales of biology. We have molecules and cells and histology, and we actually also have anatomy of an entire organ. And so more recently, we decided to take a second stab at this problem in the context of mapping the brain as part of the Brain Initiative. And here we're going to take a specimen for something like, say, single nucleus RNA-seq, but also obtain the immediate adjacent section for a histological stain. I'll show you in a little bit what this looks like. And now we would hope that we could use these two types of data along with, say, spatial measurements of molecules as a fingerprint that would let us relate our cellular profiles to spatial transcriptomics data, but also to histological image and anatomical position in something known as the common coordinate framework of the brain. For the brain, you have that. In principle, that should exist for other organs as well. And so for this, we developed a new mapping method that we called Tangram, which was developed by Tommaso Biancalani, Gabriele Scalia, and other colleagues. And it solves a non-linear optimization problem here iteratively, as you would expect. So you initialize basically by randomly distributing the single cell or single nucleus profiles in your region of interest, known as the ROI. And then we have a score that I'll explain in a second to evaluate the mapping, which we call by a Tangram loss function. And then finally, we're going to optimize with stochastic gradient descent iteratively. So really the key point of these methods is the loss function. So what does it consist of? So our function compares our cell mapping to the spatial data by considering three terms. The first is a density term. There's a density term, a gene expression term, and a cell number term. And so the first I'll describe is actually the gene expression term. And so we compare the mapped expression to the spatially measured expression for any gene for which we have such a measurement, because some genes are only measured, only some genes are measured spatially. And we do it both for each gene across the voxels and for each voxel across all genes. We also assess the cellular density term, which we do by a KL divergence metric between the mapped single-cell RNA-seq and the spatial data. And then the last piece is that we have this cell number term, which is very important under particular conditions. And so imagine that our section, the image section, has, say, 1,000 segmented cells, but our single-cell RNA-seq data might have 30,000 profiled cells or nuclei. And so we want to just select the best 1,000 cells, because that's all that piece of tissue actually allows us to have. And so for this, we have this filter F, which is convolved actually in all the previous matrices that I showed you. And so Tangram can be applied to combine single cell profiles with many different spatial modalities. So it can be data with low resolution, like in situ hybridizations, intermediate resolution, like spatial transcriptomics or Visium, and high resolution, like MERFISH. And with the different methods, we get different benefits from using Tangram. So I'll start with MERFISH. Here we're mapping the cells into the tissue. And we get this very high resolution view of which cell is where, because MERFISH itself is a high resolution method. But now we can use this projection trick to get the expression of genes that were not in the shared features. So for example, here we withheld some of the MERFISH genes during the mapping, and we can compare our predictions to the experimental measurements and see that we did quite well. Now we can, of course, predict additional genes that were not in the 150 MERFISH genes. And here we confirm them by comparing them to the Allen Brain Atlas. Now, in some cases, we compare the Tangram prediction to the measured expression for MERFISH, and they're in disagreement, but the Tangram prediction actually predicts, agrees really well with the Allen Atlas. And the MERFISH measurements basically look like a lower quality signal that does, occur sometimes in experimental techniques. And so this suggests that we can actually correct measured expression. It's less critical in a method like MERFISH, which is very sensitive, but it becomes very useful when we move to methods like StarMap, which are amazing, but they are amazing at being able to measure, say, 1,000 genes, and they pay some price for it in the quality for some of those genes. And so here, the Tangram predictions really help us correct the raw measurements from StarMap and obtain the expected layered patterns for several key genes. Now, the Tangram mapping can also be used to transfer other data to spatial coordinates when we don't have any spatial measurements for them, if we have multi-omics measurements at the single cell level. So here, for example, we use ShareSeq, which is a joint single cell and single cell RNA and ATAC-seq method that we developed together with Saima and Jason, with joint postdoc with Jason Buon-Rostro's lab. Sorry, Jason. And we don't yet have a spatial method for chromatin accessibility, although some groups work on that, but we can use the mapping to project the chromatin accessibility through the cells that we profiled, that we mapped by the RNA signal. So RNA signal we have spatially and in single cells. And once we do this mapping, the cells transport or transfer with them the chromatin data, which now becomes spatialized. And then we can compare the localization of accessible chromatin to RNA expression. We can also then do things like inferring transcription factor activity, which often actually has these beautiful localized patterns, even if the RNA of the transcription factor itself is not localized. And we're still assessing the patterns of these targets. So now, can we also use this framework to tie to other levels of organization and knowledge like histology and anatomy? And this is especially appealing in the brain because there's a lot of existing knowledge in atlases like the Allen Brain Atlas and the Big Blue Atlas, and they include many different types of features. And all of these features are tied together through histology and anatomy in what I told you is called the Common Coordinate Framework, or the CCF. So long term, we'd like to be able to see if we can just learn this CCF directly, but for now, all we did is use the knowledge that's already there. And so I promised to show you what these sections actually look like, and this is what makes it so relevant to us. And so when we planned the Atlas collection with Chuck Vanderburg in Evan McCosco's lab, we made sure that when Chuck obtains nuclei for profiling, he dissects and freezes the region, and then he takes a biopsy punch from the frozen dissected ROI for nuclei extraction and profiling, but he also sections and he stains the sections, which are actually those missing pieces that you don't see. He stains those missing pieces right above and right below, and as well as the area that actually has a hole in it, to do histology. So now we can take these histological images, which have no molecular information in them directly, and we'll want to place them into the existing atlas. And so for this, we learned a model of anatomical depth from the existing brain atlas. And we saw, and that's the model on the left, and we saw that the point distance in the latent learned space actually represented the anatomical distance between the slices. It basically became one-dimensional in the axis in which the slices are coming. And so this trained model allows us to really easily automatically retrieve an image from the Allen CCF onto which we can register our histological image. So I give you a section, you get the image from the atlas. Now, we also learned a model of the anatomical labels. Again, we use the Allen Atlas, so that now if we're given a new section, we can actually call the different anatomical regions on it with semantic segmentation, something that people do a lot. The trick for us was really to combine these two models into what would be a fully automated pipelines, despite all the imperfections and all the imperfections in the scanning the data. And the trick that we used was to actually register masks rather than histological images in the atlas directly. So for our section, we're going to do anatomical region calling to make a mask. And then for the atlas section, we're going to extract that mask, and then we're going to register masks to each other and apply this registration back to the sections. And the benefit of this is that we now can actually bring everything together into one atlas. We give Tangram our sections and single nucleus RNA-seq profiles, and we get back the anatomical regions, the cellular densities, the expression profiles of cells in each of these regions. We get all of it combined together. It also, occurred to us that it means that if we have a spatial method that has both molecular and histological data measured simultaneously, and this is the case for spatial transcriptomics or Visium, then we can benefit from this additional knowledge in the original mapping task that we did, because we can leverage the known anterior to posterior coordinate from Tangram and only map single nucleus RNA-seq profiles that were collected from punches at that particular depth. And so we leverage this together with the fact that Tangram can constrain the mapping by the observed number of cells, as I showed you. And in this way, we can deconvolute Visium, which is not actually a single cell method. So for this, we combine the probability map with cell segmentation. Then we take each of the Visium circles that you see there, one at a time, and we ask, what are the most probable cells? And this is kind of cool in terms of actually telling you these colors, but you do have to remember that the assignment within the circle itself is actually random, and that there is no one unique deconvolution. There are several solutions that seem to be compatible with the probability maps, although they in practice actually are really similar to each other. And so to conclude this second part, I showed you Tangram, which is a method that aligns single nucleus RNA-seq data to various spatial modalities. And it gives us single cell mapping, genome-wide transcript projection, multimodal mapping with multiomics data, and deconvolution. And with this additional automated registration pipeline, it lets us connect the molecules and the cells to histology and anatomy and any additional information that is out there about this histological and anatomically mapped section, which is a very rich set of information that neuroscientists have worked very hard to build. And that what gives us really a fully integrated atlas. And so for the very last part of my talk, I want to turn from these methodological questions and focus more on the ways in which we think that they can help us understand tissues conceptually, such as the roles of cells in tissues and histological modules and functional cellular circuits. And what I'm going to show you today is just a glimpse. But for us, it's really the main goal of all of these efforts. You first had to get some, decent methodologies right, but at the end of the day, what kinds of biological questions would we want to ask as we move gradually, from structure to function? And so the first and very brief illustrative example I want to give goes back to the question actually of the cell's identity. So in particular, what might happen if we define cells not just by their intrinsic features, those could be their molecular profiles or their cellular morphology, but also by their extrinsic features, for example, the cells that neighbor them or the histological structure that they reside inside. And so to learn this kind of cellular definition, we developed a model that we called in situ-vec that basically just tries to look at the cell in two ways. On the one hand, it's going to look at the cell as an image, which is not just the cell itself, but the cell and its neighborhood within some distance. And then on the other hand, it's actually going to use the cell's own molecular profile as measured in situ. It's going to encode each of them, and then it's going to sample these encodings. And then for the images, it will just decode sorry, for the images, it will just decode the images back, but for the gene expression, it's going to build a joint embedding from both the image and the expression latent spaces, from which it will try to reconstruct the expression level. And then, you know, from this in situ VEC embedding, we can perform all sorts of operations like clustering and visualization. And when we do those, we basically end up taking into account both the expression in the cell and what is going on around it as is reflected by the image. Okay. So just 1 second. Oops, I'm going the wrong way. Took me a moment to realize what was going on. And so we decided to apply to a data set that we previously collected with Peter Sorger's lab in an earlier study that measured 13 proteins in FFPE sections from 19 melanoma tumors. And just to orient you, here is one section like this. In blue, we see the T cells with CD8 T cells in light blue. In green are the malignant cancer cells that induce a program that we previously discovered from single-cell RNA-seq that's called the T-cell exclusion program. So they should promote a cold environment without T cells. And in red are malignant cells that repress the exclusion program so that they should promote a hot environment with T cells. And this is exactly what we see. There are red malignant cells that are interlaced with blue TILs, but the green cells make these big cold patches. And this is true across the patient samples, and you can quantify it and calculate the statistics, and you can show that this exclusion score is significantly higher in malignant cells that are in cold T-cell depleted niches. So this is all published work. What we wanted to see is if we can now understand these cells and their properties better by considering not just the expression of the 13 genes, but also how the cells relate to those around them. And so now we trained in situ VEC with fields of views, many fields of views from the 9 patients, and we held all the data from 3 other patients as unseen. And to see if the model generalized, especially because these are tumors, we compared the predicted expression to the measured one for each of the 3 new tumors, and I'm showing you one of them, and generally it performed quite well. And so next, we used the in situ VEC embedding in order to cluster the cells, and we included the 3 test samples, and you can see that they mix nicely with the embedding. And so now we can ask if we gain some new knowledge. So for example, for the CD8 T cells, they form 3 clusters by in situ VEC, and these 3 clusters actually make 3 distinct spatial patterns. One cluster has those T cells directly proximal to the MHC class I malignant cells. A second extreme is a cluster of T cells that make their way into the cold niche, even though quite sparsely. And we really can only see these patterns from the joint embedding. If we use only one source of features or the other source of features, we cannot make these distinctions. And then similarly for the malignant cells and the non-CD8 T cells, they form these different groupings. So for example, the Treg cells are expressing exclusion genes only in the cold zone, and they're separating the malignant cells into 4 categories with different spatial and molecular patterns. And these patterns are persistent and they generalize across the patient samples, even those that were never seen by, even those 3 patients that were never seen by the model. And so they can now suggest new categories of cells that we could not have distinguished based on the molecular intrinsic information alone, and those would require additional functional experiments to really understand what they do. But even in this example, we didn't actually look at the tissue. Still, the unit that I was talking to you about was the cell, the cell, the cell. The cells got mapped here and the cells got mapped there, and they had properties and they had categories. Can we actually go one level above that? And so for this, we're beginning to develop methods that relate the different cells to each other. And so from a tissue biology perspective, we actually expect cells to coordinate their function, either because they're all responding to the same signal, or because the cells respond to a cue that, one cell responds to the signal and it affects the other cells. And some of this could lead directly to the same response in, same genes in different cells. But even if the cells actually respond differently, which is more likely, the response should be coordinated. And so this means that we expect a signal in our data that reflects this coordination. And I'm going to call this signal multicellular programs. So if we want to relate multicellular programs and find this kind of signal, we need some way to relate the cells, the different types of cells to each other. So spatial proximity is one very obvious way to do this. And I'm going to show you that, but I'm also going to show you that you can even be looser than that, tissue locations or individuals or disease conditions. And so to look for these kinds of signals, Livnat Jerby, who just finished her postdoc in my lab and started in Stanford Genetics, being a colleague of Anshu's, she developed a two-step approach that she calls dialogue. And so the first step, in the first step in dialogue, we apply a penalized matrix decomposition approach in order to identify sparse canonical variates that would transform the original feature space, say the genes, into a new feature space where these different cell type specific representations are actually correlated across different niches or different samples. And this really uses just the average signal in different cell subsets in each niche or sample. And then we have a second step where dialogue identifies the specific genes that comprise the latent features, and it does it by fitting multi-level models. I don't have a lot of time to elaborate on this, but this is also when we use the underlying single cell distributions. And so in our first applications, we focused on niches, spatial physical niches. And here a niche is basically a pair of directly proximal cells in MERFISH data in the hypothalamus from an earlier study we did in collaboration with Xiaowei Zhuang and Catherine Dulac's labs, where they focused on parenting behavior. And so Dialogue found multiple cases of these pairwise multicellular programs. In each of these cases, it's a statistical association between the cells. Now it's again disrupting me in the middle. Can you guys still see my slides? Hopefully.
Yeah.
Yeah, thanks. I don't know why my laptop keeps doing that today. So these programs are related to different spatial distributions of the same cells. So for example, in the top are programs that couple excitatory and inhibitory neurons. And there's two different ones, MCP2 and MCP4. These are genes that are coordinated between their expression in excitatory neurons and inhibitory neurons. There's one particular pattern like that when they're organized like they are in the NCP2 example, and a different pair of programs are coordinated when they're organized like the MCP4 example. And below you see the same for astrocytes and inhibitory neurons. Then we took it one step further, and we basically decided to make the MERFISH data less good by what we call coursing it up. We dissociated it to these little regions that were about 50 to 100 microns squared in the image. That means about 500 cell aggregates. And then we learned the same MCPs with the same model, but we no longer had the immediate proximity. We just knew these 500 cells go together, those 500 cells go together, and then we use the MCPs in the expression of a given cell in the test data to predict the expression of its neighbors in this radius of between 15 to 500 cells. And we saw that actually we can make really good predictions like these from data that was essentially dissociated. And so we thought, well, in that case, we can just try it out with tissue that was dissociated to begin with and look for cell-cell relationships there. And to test this idea, we used an atlas that we recently published last year for healthy and ulcerative colitis colon. It spanned 12 healthy individuals and 18 individuals that had ulcerative colitis. And overall, and that's why we chose it, it had 115 spatially distinct samples, each of them profiled by single-cell RNA-seq. So it shows on 5 well-represented cell subsets, macrophages, 2 types of epithelial cells called TA1 and TA2, and 2 types of T cells, CD8 T cells and CD4 T cells, and looked actually beyond pairwise combinations. And so first, we wanted to see that the MCP really captures something real about the samples. So we did another little exercise. We trained the model after we contaminated the data. We took about 50 cells, 10 per type, from an adjacent sample from the same donor. So it could be a different layer or a different clinical condition. And then we used the MCPs from the trained model to predict each cell's state. And we compared it to the real state of the cell, what we actually measured for it. And we call this difference the environment score. And so using these, we could predict which cells were actually misplaced with really high accuracy based on having basically a lower environment score. We predicted them worse. And so this did vary, depends on how we contaminate. We did best when the location differed, say lamina propria versus epithelial layer. And we did the best at finding misplaced T cells and the worst for macrophages. But we think that's kind of a cute idea on how to predict unusual infiltrators in tissue. But for now, all we got from it is kind of confidence that these kinds of approaches should actually give real biology. So I'm showing you just one biological vignette. This is from the MCPs themselves. So this multicellular program is made of different genes in each of the 5 different cell types, and they're labeled on the top, the cell types. The genes are in the rows. And the genes are in the columns, but their cell type association is labeled on top. And its induction across these cells is associated with ulcerative colitis, and it's enriched with multiple genes that are located in risk loci for IBD and ulcerative colitis. But these genes are expressed in different cells. They're just coordinated with each other across these cells, across the samples. And in fact, when we score this kind of program in bulk patient RNA-seq, it's actually predictive of responses and of anti-TNF therapy. And so to conclude this final part, I showed you 2 steps that we're taking to better understand cells in a tissue context and the function of tissues: in situ vec, which helps define cells by both intrinsic and extrinsic features, and dialogue that recovers multicellular programs, which can be applied both with spatial data and across cellular samples. And I'm going to conclude the talk with this. I just want to thank the people who did the work. I highlighted them throughout the talk: Brian Cleary for compressed sensing and SciSci in collaboration with Fei Chen's lab, the work on DNA microscopy, which is really Joshua Weinstein's, and he was a postdoc between my lab and Feng's lab. The work on Tangram was led by Tommaso and Gabriele and was a nice collaboration with Evan Makosko and Zhao Zhuang. On in situ vec by Ishit, and the work on dialogue by Livnat Jerby, who's now in Stanford with many people on this in this meeting. And I'll be happy to take any questions. I apologize for the many technical problems with my laptop, but they seem to have bothered mostly me. Thank you very much.
Thank you so much, Aviv. That was great. Let's just jump to questions. There are a few questions trying to pop up in the Q&A. So the first question is from Omid Bazgir. He asks, I think that he's referring to your the first part, the compression part of your talk. Could you please clarify why you do SVD and then NMF?
No, we don't do SVD and then NMF. We compared 3 methods: SVD, sparse NMF, and SMAF. So it's 1, 2, 3, but not first do 1, then do 2, then do 3. So I guess the slide was a little confusing on this. So we compared these 3, and we show that SMAF performs better for decompression than these other 2 alternatives. That's all. And I didn't show the details for that. This is long published. Yeah. Sorry for the confusion.
Thank you. Next question is from Brody. Asks, thanks for the great talk. How critical do you find the architecture and hyperparameters of the autoencoder to performance? Did the decompression work using a cursory attempt, or did it require a huge amount of experimenting and fine-tuning?
So I wouldn't say a huge amount of experimenting and fine-tuning. I will point out that maybe it was more painful to Brian than it was for me, but it didn't take a lot of time and a lot of fiddling. So I think that's still a very fair statement. I will say, and you can see it actually, we updated the bioRxiv posting recently, so you can see kind of the latest version of this story. It's been out for a while posted. I will say that there are cases when the autoencoder, when we prefer the segmentation-free method, and there are cases where we actually like the segmented method. So we had a hard time at the end of the day saying we obviously favored this over that or vice versa. I highlighted this one because it was unusual, and I thought that it's always good to notice the alternative possibilities. But in the cases when it does or does not work well, it's really about the decompress. As you decompress, there's harder and easier things to solve. Some genes behave much better than others. Some of it is also the limited number of combinations that we had that requires all sorts of post hoc little solutions that get better and better the more combinations you have. So some of the performance of the different algorithms is more related to that than I think to the fundamentals of the problem. But generally, I wouldn't say a very difficult one, as these kinds of fine-tuning experiences go.
Great. Maybe we have just time for maybe one more question. Anyone from the panel? Yeah.
I have a question for Aviv. Sorry. Thanks, Aviv, for the great talk. That's really fascinating work.
Hey, Jane.
I was wondering if you could maybe comment a little bit more on, especially given your new perspective in your new position of how the spatial and tissue information that you're learning could be used for therapeutics and drug discovery. Like, what do you think are some of the most promising applications?
Oh, I think there's many promising applications, and some of them you will see faster than others usually for these domains, given where you are. For example, in a making medicine drug discovery process, there could be things with huge impact, but if they're applied to the very early steps of the process, you're going to see the payoff in 10 years from now. And if you're applying them to things that are done very late in the process, you're going to feel the payoff faster, but it doesn't mean that the payoff is bigger. So the temporal and the amplitude and the extent of immediate gratification are often anti-correlated in that domain. It's not for people who need fast gratification, but the amplitude of the gratification is extremely high at the end. That's a little bit how I try to describe it to people. But so I'll try and describe from two perspectives. The first is things that actually happen relatively late. One of the challenges that you always have when you develop medicines is really understanding what is happening in the patient's body. So in a tumor, this would be particularly meaningful because you can obtain a lot of specimens from patients and you can look at them. And we're often limited in looking, especially in interpreting what we see based on knowledge that we already have. So for example, imagine that you're using your favorite deep learning algorithm to do your most fantastic thing. At the end of the day, if you pose to people the things seen, they still need to perform an act of interpretation in order to understand what has been going on. So if it's just for prediction purposes, it's one thing, but we usually need to understand why in biology. It's kind of not enough for us to just say this predicts that. We want to understand why and how. That's what makes biology a little different than other domains. And so for that, the ability to put together these different layers of biology into a single model is tremendously impactful because the molecular level tends to be a lot more interpretable for us. But the histological level carries a very substantial richness, as I showed, both on how the tissue actually functions, who is doing what to whom and how, but also the legacy knowledge of this is what we call this, and this is what we call that, and this is what we've seen here, and this is what we've seen there. So I think that is one domain that is active and is going to expand a lot and has a lot of impact. The other piece is when you're starting to think about how would you actually develop new medicine. In the end, disease happens in tissues. There's impact, right? So think even on a very genetic setting for a disease, say, a large impact single gene kind of genetic disease. It impacts within a cell, which resides within a tissue. And a lot of what you see at the end is the tissue effect. If you think about a common complex disease like IBD or ulcerative colitis, which was in my very last example, actually the risk is distributed across many cells in the tissue, and they're going to impact each other. We don't understand that very well. And when we come with our interventions, it reflects our limited amount of understanding. And so the other place where I think we have a fantastic opportunity is to not just do interventions, both in models and by looking at a diversity of human specimens. Basically, both of these are interventions. Either we do them or nature do them. Not to just do the intervention that we know would have an impact in the tissue, but to actually monitor their impact in these terms, rather than try to reduce everything just to the individual cell level and the individual genes inside the individual cell. And so things like screening in vivo in this way, and using natural genetic variation or mutations in cancer, combined with rich spatial information, is going to have a dramatic impact on the types of, on how we define targets and interventions for disease, which is what gives us medicines at the end. I would say these are probably the two biggest ones. There's a lot of specific additional things, toxicity, and assessing toxicity is a very big one, and is usually assessed at the tissue level and other applications like that.
Great, thank you.
Thank you so much, Aviv. So in the interest of time, I think
Thank you, everyone.
It was a great talk. So we'll start off with the selected speakers now. The first speaker is Manu Saraswat, and he will be presenting a talk on convolutional additive models, a fully interpretable approach to deep learning in genomics. Manu, if you're there, can you start your video and share your screen? I actually do not see him in the panelists list. see if he's in the attendees. Oh, I do. Okay. Let me move him. Manu, can you hear me?
Yes.
Okay, great.
Can you see my slides?
Yeah, can you start sharing your slides and also your screen?
Yes. Awesome. Can you see my screen?
Yes.
Okay, cool. Hello, everyone. My name is Manu Saraswat. I'm currently a first-year PhD student with Oliver Stegle and Moritz Mahl in Heidelberg, Germany. But today I will be talking about my work in Vancouver titled Convolutional Additive Models, a fully interpretable approach to deep learning in genomics. This was done with German Novakovsky, Etienne Munier, Uriel Fornes, and supervised by Sarah Mustafavi and Wyatt Wasserman. So deep learning methods have become pervasive in biology and healthcare, and they are being used in different domains, such as for analyzing electronic health records, and analysis of medical images, drug discovery, and understanding genomics. Particularly for genomics, deep learning models have been used at different levels of gene regulation, starting from 3D organization of the genome to understanding DNA accessibility, DNA methylation, binding of transcription factors, and so on and so forth. The recent advances in deep learning in genomics have been driven by convolutional neural networks. Now, these methods take a one-hot encoded DNA sequence as input and predict a functional output, such as binding of transcription factor from, say, ChIP-seq data or chromatin accessibility from ATAC-seq data. The fundamental unit of such neural networks is a convolutional filter, which can be thought of as a position weight matrix. The model consists of multiple such filters, and the CNN learns non-linear combinations of these filters together to make the prediction. Now, these approaches outperform traditional methods based on k-mere frequency, and they do not require handcrafted features, but it's difficult to interpret these methods. It's useful to have some notion of interpretability in the deep learning models we develop for two main reasons. First is that if we understand how the model makes its predictions, we have more trust in the model, and then we can use those models in unknown settings. Also, it will help us to debug the model if it's learning something stupid. So over here, an example is shown where a model was developed to detect pneumonia from chest X-rays. And when the model was used to do these sort of predictions in images from a different hospital, it failed. And the reason was that the model basically learned the hospital token as one of the features. And when it was used to predict on new hospital data, it failed. The second reason why we need interpretability in deep learning is if we are able to understand how the model is making its predictions, particularly in biology, it can lead us to novel insights. So an example here is shown from the Bpnet paper, where deep learning models and interpretability suite of DeepLift and DF-Modisco was used to decipher cooperativity between transcription factors and understand soft syntax of their binding. Now, I'll briefly talk about the existing methods of interpretability in sequence-based deep learning. The first method is convolutional filter-based method. So we represent convolutional filters as motifs of known transcription factors. Then iteratively, we can nullify these filters and look at their impact on the model performance. And if it leads to a large change in prediction, it means that this filter is perhaps important for the model. And in this way, we can link the importance of a known transcription factor with, say, predicting accessibility in a particular cell type. However, there are several drawbacks associated with this method. Firstly, the filter representations we learn this way are dependent on architectural choices, particularly the length of filter, whether we have used, what is the size of max pooling we have used, and the type of activation functions we are using. Also, identifying how different combinations of filter affect the prediction is time intensive, because that would basically involve nullifying like combinations of 2 or 3 filters at a time. And thirdly, this method does not explain how the prediction is being made. The other method for interpretability is using feature importance scores. So once your model is trained, these methods give insights into which particular positions in an input sequence are important for making the prediction. So a simple way of doing it is using perturbation-based methods or in silico mutagenesis, where you would basically mutate each position in your input sequence and look at the prediction, whether it's changing or not, and accordingly decide whether a position is important. Or we could use backpropagation-based methods such as deep lift and GradCam to achieve the same task. Again, there are drawbacks associated with this method. Firstly, these methods might not provide reliable scores in all settings. And since we achieve these importance scores at the level of individual input sequence, it's hard to quantify their global importance. And third, again, like the previous method, they do not explain how the prediction is actually made. Now, to overcome these drawbacks, we propose our method called Convolutional Additive Model, or CAM. Now, CAM consists of several independent units. Each of these units consists of a single convolutional filter, followed by 2 fully connected layers. Now, since we have, say, n of these units, and when we want to make a prediction for, say, binding of a transcription factor FOXA1, we would just do the weighted sum of outputs from each of these individual units. Now, by just visualizing the final layer of our model, we get insights into which units are important for each of these outputs. So over here, for example, if we want to look at which units are important for JUND, we would just visualize this final layer and see that filter 0 and filter 8, for example, are important for JUND. And so the global quantification of importance of these units becomes trivial just by visualizing. We don't have to do any nullification experiment here. Now, the idea for this model was like inspired by the recent paper of neural additive models. What the authors in the paper did is, instead of using a usual feed-forward neural network, which basically takes all the inputs, then everything goes into everything, you have these fully connected layers. What the authors instead did was, for each unit, they had a separate neural network. And the final prediction was basically a weighted sum of outputs from each of these different units. And the authors showed that on a bunch of different tasks, both these models perform same, even though the NAM model, the neural additive model, seems to ignore the nonlinear combinations between the inputs. So as a proof of concept, we used the CAM model for prediction of transcription factor binding of 3 transcription factors, FOXA1, CTCF, and JUND, based on ChIP-seq data. And we took a 200 base pair long one-hot encoded DNA sequence as input, and our CAM model had 10 independent units. And to ease the interpretability, we forced the final layer weights to be positive. Now, looking at the results on the left, these are the precision recall curves and the ROC curves. And the AUCs of these curves are pretty decent, and they're comparable to what we got when we trained a multi-layer CNN with 3 convolutional layers, the values for which are shown in the brackets. But what we're interested more is in interpretability. So we visualize the weights of the final layer and also converted the filters into PWMs. And we found that, for example, in the case of JUND, filter 0 is important, and that filter in the model actually corresponds to the motif of JUND. And similar observations were made for FOXA1 and CTCF. Now, one limitation of filter to PWM conversion is that we might not get meaningful motif representations. So to overcome that, instead of converting filter to PWM, we used each individual CAM unit as an oracle and generated synthetic sequences which maximize the output. So the idea is that since we focus on this entire unit and not just the filter, those sequences which maximize the output from this unit can lead in insights into what this unit is actually learning. So for our experiments, we use genetic algorithms to generate sequences which maximize the output. In the previous example of TF binding prediction, we saw that filter 1 is actually important for CTCF binding, but it does not it does not correspond to any meaningful motif representation. So we use genetic algorithms to make synthetic sequences which maximize the output. And we found that those sequences contained motifs of C2H2 zinc finger proteins. And it makes sense that why this particular unit could be useful for prediction of CTCF. Then to test the capabilities of accuracy prediction and interpretability of our models, of our CAM approach, we resorted to a more complex task of predicting chromatin accessibility in mouse immune cell types. So we use ATAC-seq data from 81 different mouse immune and stem cell types. We took the open chromatin regions from this data set, took the one-hot encoded DNA sequence of each of these open chromatin regions, and predicted their chromatin accessibility in all the immune and stem cell types. And highlighted in yellow is the B cell lineage, which we'll discuss in detail. And we used a CAM with 300 units to make this prediction. And the performance of the model was measured as a correlation between the predicted and actual accessibility. And as a comparison, we used the AI-TAC model, which was recently published and was a deep learning-based analysis of this data set. So looking at the results, the average correlation of the test set is very close to what was achieved using AI-TAC. Then we converted the filters of each of these individual CAM units to see what they are learning. And several of them correspond to known regulators of immune cell differentiation, such as PAX5, EVF1, and F-kappa beta. Now, the most interesting part is that we wanted to see that whether we can assign these transcription factors to their particular importance in certain cell types. And for doing that, we just visualize the final layer of our CAM model. And this is what we see. Now, the rows here represent the different cell types, and the columns are the CAM units. And each value is the particular weight for that particular cell type and the unit. And focusing here on the B cell lineage, we see that there is a group of units which is particularly important for B cell chromatin accessibility prediction. And looking into detail, we see that this is actually PAX5 and EBF1 motifs. And we get similar cluster over here for myeloid cells and a cluster for stem cells as well. Now, if we were to generate this heat map using traditional methods of nullifying filter, we would have to take each filter, nullify it like for each of the 81 cell types, and then get this one entry. So it will be much more time intensive, whereas with our method, it's just trivial to visualize the final layer. And this particular group of transcription factors important for stem cells was actually not recognized by the AI-TAC model. This is something we recovered with our method. So in short, we make the interpretability very easy, and the performance of our method is also comparable to the multi-layer convolutional methods. So now the question is that on the face of it, CAMs ignore the higher level interaction between filters because they are independent, and yet they achieve similar level of performance as multi-layer CNNs. So what is happening there? So the only difference between the CAM approach and our approach and PWMs is the use of fully connected layers. And we know that PWM methods do not perform really well. So we wanted to interrogate the role of fully connected layers. And to do that, we used the method employed in the Bpnet paper. So we focused on one single CAM unit. We generated importance scores from deep lift. Then we clustered those seclets and generated a consensus motif for each of these CAM units. And then we inserted these consensus motifs into random sequences and moved them along the sequence to see if there is a positional importance learned by these fully connected layers. And we see that there is some positional information learned, because if the motif is present in the center of the sequence, there is an increase in the model prediction, whereas on the sides, there isn't a lot of change. Now we compared it to a model where we remove all the fully connected layers and just use simple max pooling. So for each of these filters, we have just one output, which is basically the maximum activation of this filter in the sequence. And when we repeated the exercise, and obviously, since there is no positional information here, we get the same level of activation at all positions. But to our surprise, the model still performs as good as the one with fully connected layers, like similar average correlation on test set and same level of interpretability, which is surprising because we have lost all positional information and all higher level interactions. So the question is, do we really need positional information and higher order interactions for modeling regulatory activity of TFs? And why do deep learning methods perform better than PWM approaches? Is it because they learn better motif representations? Are they learning new motifs? Or is it just that the level of the data modality resolutions we have right now are, the simple methods are just enough to model them? And with this, I'll conclude my talk. So the three conclusions: CAMs achieve global interpretability through final layer weights visualization without compromising on accuracy. Using synthetic sequences, CAM overcome the limitations of filter to PWM conversion. And CAMs recover regulatory landscape of mouse immune cell differentiation without time-intensive interpretation techniques. Thank you, and I would be happy to take questions, comments, or suggestions.
Great job, Manu. Fantastic talk. So I'll quickly go through questions. We've got quite a few. So the first question is from Ben Zhao. Is the CAM strategy limited to a single convolutional layer? Have you tried extending it to 2 or more convolutions for each additive neural network to capture higher level filter-filter interactions, of course, at the expense of runtime and some interpretability?
We actually, the thing is, just with using single filter, we were able to achieve what multi-layer CNNs do. So we didn't feel the need to try that approach. And as you said already, that if we use multiple filters, that will come at the expense of losing some interpretability. So we didn't do it. But what we did try is compare a single convolution layer approach. And it's interesting that a single convolution layer approach does not work, but the CAM approach does work, which has independent units, like single filters. So yeah.
Excellent. Next question is from Alex Liu. Great talk. Does this kind of architecture only work for losses that use a single score? Or do you think they can be adapted to autoencoders or generative self-supervised outputs?
I think they should work for other architectures as well. I don't see why that would be a limiting case for it. The only thing we are doing is instead of learning higher order interactions, we are just separating them into and making a very additive, simple version of it. So I think they would work for those cases as well.
Great, great. Sean Mahoney asks, if the fully connected layers are not adding anything to the performance, then does CAM outperform a linear combination of PWM-based methods or an SVM, for example?
Yes, thanks for asking this, Sean. And we actually tried this, and I have a slide to show what happens. So this is the correlation on test set we get with PWM-based approach. So this is slightly less than what we get with the usual model. And on the right is the interpretability, the matrix. It's not very clear. So we do get some information, but it's not as good as the full CAM model. But like it's an interesting exercise to see that, because the fully connected layers don't really add anything. We see that. So I actually looked at in the literature and found a few papers which used PWMs with dinucleotide frequency and feed it into a logistic regression, which is basically what our final layer is doing. And this approach basically outperformed the DeepC based architecture. So what I haven't tried is PWMs with dinucleotide frequency, and maybe that performs as good as the multi-layer CNN or the CAM. So that is something I would want to try and see.
Excellent. Let's see, there's lots of questions coming up. Do your results suggest that there are no or very few spatial relationships between different motifs in regulated regions?
That is a question which I left as an open question to all the audience as well, because on the face of it, it looks like that indeed, without including any spatial interaction, we are still able to achieve what you would get with something which includes those interactions. So yeah, our model.
That's exactly 20 minutes. Thanks so much. There are a ton more questions in the Q&A, so it'll be great if you can go in there and just answer them.
Okay, sure, I will do that. And thanks everyone for listening and for the questions.
Excellent, thank you. I'll shift this over to now Sarah, who will host the next talks.
Okay, hi everyone. This is Sarah, the other one of the 5 co-organizers. So we're going to move on to the next talk by Avanti Srikumar from Anshul's lab, and she's going to talk about benchmarking reverse complement strategies for deep learning models in genomics.
All right, thank you. Yeah, can you hear me and see my slides and everything?
Yes.
Okay, awesome. All right, great. Well, hi, I'm Avanti, and I'm excited to talk to you about our work on benchmarking reverse complement strategies for deep learning models in genomics. This is joint work with my mentee, Hannah, who's a very talented high school senior, in case anybody's like on college application panel, she's excellent, and my PhD advisor, Anshul. Okay, great. So in this work, we're concerned with models that predict signals of regulatory activity given DNA sequences input. Our labels come from experiments that measure genome-wide regulatory activity. For example, a ChIP-seq experiment produces a signal that indicates the binding strength of a particular transcription factor at each region in the genome. The inputs to our models are DNA sequences, and the outputs predict the experimental signal measured at those input sequences. And there's many types of output prediction tasks. So in this work, we'll be concerned with binary models, which predict the presence or absence of a signal peak over the input sequence, and profile prediction models, which were mentioned in the earlier talk, and they predict the shape of the signal along the input sequence. And our models will be convolutional neural networks, or CNNs, which, as you saw, tend to give state-of-the-art results when applied to these types of prediction tasks. Okay. So here's a depiction of a CNN architecture. Sorry, is there some echo or some issue?
No, go ahead. Just
okay.
mute it, somebody else. Yeah.
Okay, cool. So here's a depiction of a CNN architecture. The input to the model is the DNA sequence represented as 1s and 0s, and the layers in the CNN learn patterns called motifs that transcription factors like to bind to. For example, the sequence shown here contains a match to the GATA motif. And because double-stranded DNA has complementary base pairing, we should expect that the model will give a similar output, even if the reverse complement sequence is supplied as an input. Unfortunately, standard CNN architectures are not guaranteed to learn the symmetry. So although the model may have learned to detect the GATA motif in the forward orientation, it may not recognize the motif when it appears in the reverse complement orientation. And the difference between strands can be quite large, even when the training data set is augmented with reverse complement sequences, as shown in the scatter plot from a previous paper. So 2 strategies have emerged in the literature to guarantee reverse complement symmetry. Prior to this work, these strategies had only been proposed for binary or regression prediction models. That is, they hadn't been extended to profile prediction models. So I'll first describe how they work for binary models and then talk about how we extend them to profile prediction models. The first strategy feeds both the forward and reverse complement versions of the input sequence through the same model, and it combines the predictions to form the output. So these are called conjoined or Siamese architectures. And note that the merging of the output across the forward and reverse complement strands can be performed either during model training or after the model has been trained, which we call a post hoc conjoined model. And so far, post hoc conjoined models have not been benchmarked systematically against trained conjoined models. And the main drawback of conjoined models is that the convolutional layers must learn the forward and reverse complement versions of each motif as though they're two completely separate motifs. So to address this, another strategy that has been proposed is reverse complement parameter sharing, or RCPS. And this strategy was first proposed in 2017, and it has been applied and extended in multiple published papers. So to understand how RCPS works, we'll first make an observation about how we take the reverse complement of a one-hot encoded sequence. And we'll assume we've done the one-hot encoding using the ordering ACGT in the channel dimension. So in this case, taking the reverse complement involves flipping the one-hot encoding along the length dimension, which corresponds to the reverse in reverse complement, and also exchanging the paired bases, that is exchanging A with T and exchanging the paired bases, that is exchanging A with T and G with C, which corresponds to taking the complement in reverse complement. And note that when the ordering is ACGT, this is equivalent to flipping the sequence along the channel dimension. So in RCPS, this idea of complementary pairing is extended to the convolutional layers. Specifically, each convolutional filter's weights are paired with those of a new RC filter in such a way that the RC filter recognizes the reverse complement of whatever pattern its partner filter recognizes. And as a result of this pairing, if we were to flip the layer activations along the length and channel dimensions, we would get what the layer activations would be on the reverse complement input. And so we achieve what's called equivariance in the convolutional filter activations. Specifically, if we define the revcomp operation to mean flipping of both the length and the channel dimensions, then taking the revcomp of the layer activations is equivalent to computing the layer activations on the revcomp of the input. And if you find it easier, you can also think of the revcomp operation as a rotation by 180 degrees, which is equivalent to flipping the input along both the dimensions. The pairing of the forward and reverse complement filters exists not just in the first convolutional layer, but also in the higher convolutional layers. And as a result, the equivariance property is maintained on higher convolutional layers. And finally, in order to make a prediction that combines the information from both strands, the forward and reverse complement channels are merged prior to being supplied to the fully connected layers. So as mentioned, neither conjoined nor RCPS architectures have been extended to base pair resolution signal profile prediction models. And what's more, RCPS models have not been systematically benchmarked against conjoined models. And even among conjoined models, it's unclear whether models that are conjoined during training outperform models that are converted to conjoined models after training. So in this work, we address these three limitations. Let's begin by discussing how we extend reverse complement strategies to base pair resolution signal profile prediction models, or the so-called Bpnet models proposed in Avsek et al., which was mentioned in the previous talk. These models learn to predict the shapes of TF footprints at base pair resolution given DNA sequences input, and they can do an extremely good job. So here are some example observed profiles for OCT4 and SOX2 chip nexus data, and here are the corresponding predicted profiles. And these profile predictions are so good that for some tasks, they are closer to the original single than even biological replicates. And the model can also learn many subtle features of the regulatory code that are missed when you use standard binary or regression models. So let's delve into the architecture of a standard Bpnet model. The goal of these models is to predict for each strand what fraction of the reads in the output window were observed at each base. Bpnet architectures are still convolutional neural nets with some subtleties such as dilated convolutions for larger receptive fields and skip connections to increase the depth. And the main difference in these architectures is the output layer, which is a convolutional layer that has two channels, one per strand, and a length equal to the size of the window that we want to predict the output over. A softmax nonlinearity is applied to each convolutional channel to get the final predicted probability distribution for each strand. And this predicted probability distribution is then evaluated using a multinomial loss function. And in order to account for sequence-specific biases in the assay, a control signal track may be concatenated as an extra channel at some intermediate convolutional layer and supplied as an input to the later layers. So let's talk about how we can make a conjoined version of the Bpnet architecture. As before, we start by applying the model to the original input sequence. However, we'll stop short of applying the softmax operation just yet, so the outputs will represent the logits of the softmax for the positive and negative strand. And as before, we then reverse complement the input sequence and supply it in parallel to the same model. And the control signal track is concatenated as an extra channel of an intermediate convolutional layer and would also have to be reverse complemented to match the orientation of the input sequence. In our models, the control signal track is not stranded, so reverse complementing it is the same as reversing it. And I've added some toy values to make that clearer. After the control track is concatenated, we can continue with the model until we get the predicted logits. And here we have to note a subtlety. The predicted logits for the positive strand, given the reverse complement inputs, actually corresponds to the predicted logits for the negative strand, given the original inputs, while the predicted logits for the negative strand, given the reverse complement inputs, correspond to the predicted logits for the positive strand, given the original inputs. And so to get our predictions on the reverse complement to match our predictions for the original input, we need to again apply a reverse complement operation to get the predictions. We need to apply a reverse complement operation to the predictions, which as before consists of flipping them along both the length and the channel axes. And then once the predictions line up, we can safely add the predicted logits together and then apply our softmax operation to get our output. So what about RCPS Bpnet? As before, each convolutional filter is paired with an RC filter that recognizes the reverse complement of its partner. And this property gives rise to the reverse complement equivariance that we discussed earlier. But what about the input control signal track, which is concatenated as an extra convolutional channel? Well, in order to maintain the reverse complement equivariance, this track is simply paired with its reverse complement, which is concatenated to the other end of the convolutional filter stack. Finally, instead of having two completely separate output channels for each strand, the weights of the two channels are paired using the reverse complement weight sharing. And as before, a softmax is applied to each strand in order to get the final signal profile shape predictions. This setup maintains the reverse complement equivariance property throughout the network. So now we come to the crux of the paper, which is the benchmarking. We use two types of datasets, binary and profile prediction datasets. Our binary datasets are similar to the ones used in our original RCPS paper from 2017. We had 3 TF ChIP-seq datasets from ENCODE, where the positive set consisted of 1 kilobase regions around high confidence ChIP-seq peaks, and the negative set consisted of accessible regions that didn't have high ChIP-seq signal. And we also had 2 simulated TF binding datasets that differed in the length of the sequences. That is, they were 200 base pairs and 1 kilobase pairs, respectively. And which contained motif instances that were sampled from 3 different PWMs. For base pair resolution signal profile prediction, we used the ChIP-Nexus data from the Bpnet paper by Avsek et al., which contained 4 TF datasets. And the base pair resolution signal was predicted for 1 kilobase pair regions surrounding the ChIP-Nexus signal peaks. And this gives a total of 9 datasets. And for each architecture and dataset, we trained models with 10 different random seeds and early stopping to assess performance. So one of the key takeaways of our benchmarking was that post-hoc conjoined models outperform models that were conjoined during training. As a reminder, post-hoc conjoined means that the model is trained normally and the predictions on the forward and RC strands are combined at test time. And note that the training dataset is augmented with reverse complement examples. By contrast, conjoined during training means that the predictions on the forward and reverse complement strands are combined while the model is being trained. And as you can see, the purple bars are consistently comparable to or higher than the red bars across all 9 datasets. And the other 3 bars are green for RCPS, orange for standard models trained with reverse complement data augmentation, and blue for standard models trained without reverse complement data augmentation. And just as a point of clarification, there's no need to do it's redundant to do reverse complement data augmentation for RCPS and Siamese architectures because that is mathematically equivalent to just duplicating your input examples because they already see the information implicitly see the information from both strands. Okay. So here's a mathematical intuition for why post-hoc conjoined models tend to do better than models that are conjoined during training. Imagine a standard model that we denote with M that is in the process of being trained. And let's say M overpredicts on a sequence S while underpredicting on its reverse complement. If the training data set is augmented with reverse complements, then S and its reverse complement appear as separate examples in the data set. And the gradient update during training would try to lower the prediction on S while raising the prediction on its reverse complement. Now consider the case where M is conjoined during training. Here, the predictions on the forward and reverse complement are combined, for example, by averaging. And thus, the prediction might be close to the true value and may not be updated during training, even though the model's predictions are incorrect for each strand. For example, as to get a sense of why they may be incorrect, what may be happening is perhaps the model is missing a motif on the reverse complement strand, and it's canceling out that with some artifactual signal that it's recognizing on the forward strand. And as a result, if you have models that are conjoined during training, they're less likely to converge to the most generalizable solution compared to models that are simply converted to conjoined models after training. In the latter case, it's more like ensembling your predictions. So another finding from our benchmarking was that the performance of RCPS is inconsistent across data sets. Although RCPS models sometimes achieve the best performance, there are other tasks where RCPS does, sometimes it's the best performance, but there are other tasks where it sometimes does quite poorly compared to the other models, particularly in the case of base pair resolution signal profile prediction. And this poor performance of RCPS is a bit unexpected, because as we prove in our paper, the RCPS architectures can represent any solution learned by the conjoined models. And thus, if the RCPS models are failing to outperform the conjoined models, it's because they've converged to a less than ideal solution. So I'll give a sketch of how the proof works. As a reminder, in the conjoined models, the model is run in parallel on both the forward strand and the reverse complement strand, and the model predictions are combined to produce the output. So we're going to illustrate how the layer activations of the conjoined model can map onto the layer activations of an equivalent RCPS model. What happens is that the activations of the conjoined model on the forward strand become the forward filter activations in the RCPS model, while the activations of the conjoined model on the reverse strand, or reverse complement strand, become the RC filter activations in the RCPS model after applying a reverse complement operation. And finally, the act of combining the predictions across the strands in the conjoined model maps onto the step where the forward and RC channels are combined in the RCPS model to give the final output. So to summarize, in this work, we extend RC architectures to signal profile prediction and performed systematic benchmarks. We found that post-hoc conjoined models consistently did as well or better than trained conjoined models and present a mathematical intuition for why. We also found that the performance of RCPS was inconsistent across datasets. So although it sometimes performs the best, it also sometimes does poorly relative to other architectures. And this happens in spite of the fact that the RCPS architectures are capable of representing the solution learned by the conjoined models, which means that the RCPS models are not converging to the ideal solution. And so we recommend that users who are interested in RC symmetry should actually default to post-hoc conjoined models before exploring RCPS. All right. Yep, that's all I have. So thank you.
Thank you so much, Avanti, for a great talk. You have a number of questions. We'll see how far we can get. So starting from Lai Shen, why not use conjoined in both training and post-hoc?
So they're kind of in, maybe I'm misunderstanding the question, because they're kind of in opposition. Like, sorry, okay, maybe, okay. If the model is conjoined during training, we also retain that conjoined property when the model is deployed. So the models that are conjoined during training are also, they're also retain that conjoined aspect when they're deployed. It's not that we take it away when we deploy it. So the only difference is whether the model was also conjoined while it was being trained.
Cool. Okay, so next question. My understanding is that some TFs are strand specific in their binding and activity while others aren't. How prevalent do you see your symmetry architecture in the wake of existing and strand specific specificity for a subset of TFs?
So So my understanding, when you say strand specific, DNA is inherently double-stranded. So maybe the TF is like binding in a particular orientation, but when it comes to like recognizing the motif, like the signal that we get from ChIP-seq, it's just saying like, you know, that you have a TF binding in this area, and the motifs that we pick up, the motifs are just saying like, this is a motif from like the ChIP-seq signal. It's not telling you about the biophysics of how the TF is binding. That motif, we should be recognizing it, whether the motif occurs in the forward or the reverse strand, because the DNA is inherently double-stranded. And of course, like if you have something that's binding to single-stranded like RNA, of course you would not want to use this reverse complement symmetry. But the DNA is inherently double-stranded, so we should recognize the motif whichever strand it occurs on. I hope that answered the question. Yeah.
Great. One more from Jacob. Thanks for the great talk. In case of conjoined models for binary predictions, you can sum average the forward and reverse complement representations before passing them to the fully connected layer. Have you compared post-hoc conjoined models with multiple fully connected layers to analogs conjoined during training? Do you think representation merging before the fully connected layer could impact the performance in those cases?
So in the conjoined models, so in the architectures we happen to train here, we didn't have multiple intervening fully connected layers, but more generally in the way that conjoined models are described in the paper, that merging is actually done to the logit of the sigmoid. So if you had multiple fully connected layers, that merging would be done, you know, towards the end. And if you wanted to like do the proof for like how that would map onto an equivalent RCPS architecture, then you would basically have the equivalent of like RC fully connected units, which, you know, it's a trivial extension of the way you would do it for convolutional units. So I may have like missed the nuance of the question. So it could certainly matter where you do the merging of the strands if there's like, okay, so yeah. When it, okay, maybe this is the question. When it comes to like when you should merge the two strands, for most regulatory genomics like applications and signals, the sequence that we provide is not oriented in any particular way. So we expect that, you know, if the output is strong when a motif is located 10 base pairs left of the center, it should also be strong if the motif is located 10 base pairs to the right of the center, which means that any positional patterns we learn should be roughly symmetric. But if in an application where that's not the case, where maybe you've like oriented it, you're dealing with promoters and you're always orienting your sequence such that the transcription start site is to the right, then in those applications, it could certainly be a bad idea to do the merging of the Siamese architectures or the conjoined architectures prior to the fully connected layers, because the fully connected layers may learn some asymmetric positional pattern. So you could certainly do the merging later, and there's, yeah, this framework allows you to do the merging later, you know, up until the way that we've written the code, the merging is done at the logit. Just prior to being provided to the sigmoid.
Okay, thank you so much, Avanti. The questions are still in the QA, so you can go and check if you want to, because some of them were complicated. And there's a few other questions for you, so if you have time to answer those. Okay. Thanks again so much. So we can go to the next talk. That's by Dan Lee, and he's going to talk about detecting higher order structural changes in 3D genome organization with multitask matrix factorization.
Cool. Thank you. Can everyone hear me and see the slide and also see my mouse that I'm waving in the screen?
Yes to all. Okay.
Thank you. All right, we'll get started. Okay, hello everyone. My name is Taiyin Lee, and today I will present our multitask matrix factorization method to detect higher order structural changes in 3D genome organization. So the human DNA, if you stretch it out linearly, would form a chain about 2 meters in length. So this long molecule storing our genetic code is packed into a cell's nucleus whose diameter on average is 6 micrometers. So this level of compaction is equal to or is equivalent to folding a 24-mile long thread into a tennis ball. So in order to accomplish this feat of compaction, the DNA is tightly wound around histone proteins, and various other proteins alternately fold, unfold, and bind to the DNA, leading to a very specific control flow of our genetic code depending on the state and the function of the cell. And we refer to this systematic packaging of the DNA as 3D genome organization. And it has emerged as a key regulatory mechanism behind cellular processes. So one way it can influence function is by bringing regulatory regions like enhancers and promoters together in 3D space, triggering gene expression. Hi-C, which stands for High-Throughput Chromosomal Conformation Capture Technology. Now, this technology enables the study of 3D genome organization by measuring the tendency of genomic regions to interact. It generates a symmetric matrix like this, whose entry represents how often the region corresponding to the row interacts with the region corresponding to the column. So here we see as an example, the Hi-C interaction count matrix capturing the interaction between an enhancer and a promoter region, which are pieces of DNA sequences with a control function that can, again, trigger gene expression. So the analysis of Hi-C data has revealed that the genome is in fact organized into higher order structural units, for instance, like chromosomal territories, where each chromosome tends to localize in the nucleus, and compartments, with A compartment being the open, accessible domains enriched in transcriptionally active regions, and B compartment being kind of the coiled up repressed regions. And we also have local structures like topologically associating domains, or TADs, which are preferentially interacting neighboring regions. Now, TADs are enforced by architectural proteins like CTCF and the cohesin complex, or the transcription elongation activity by the RNA polymerase. Now, these organizational principles and properties are conserved across many species, including mammalian systems. And the dynamics of 3D genome organization is of particular interest. It has been shown that the 3D genome changes throughout the differentiation process, as well as during pluripotency reprogramming. And disruptions to 3D genome across multiple structural scales can lead to cellular malfunction. For instance, large chromosomal rearrangements or structural variants deviating from the normal karyotype are hallmarks of many types of cancer. So here we see, for an example, a Philadelphia translocation, which leads to the BCR-ABL gene fusion and is used as a kind of a diagnostic marker for chronic myelogenous leukemia. And here in a breast cancer cell line of T47D, we have a Hi-C data visualized at a heat map here. We see the disrupted chromosomal interaction pattern when it's compared to a normal, quote unquote normal, human mammary epithelial cell at the bottom. Even at finer local scales, structural disruptions can cause havoc. So here we see how an inversion, sequence inversion around a existing TAD boundary brings a previously insulated enhancer, which is this tiny orange oval here, right next to the WANT6 gene, and this triggers some kind of a gene regulatory cascade program, leading to the F syndrome syndactyly. So a key problem in regulatory genomics is to detect higher order structural changes in dynamic processes and across diverse contexts. And there are existing computational methods to detect changes in 3D genome organization. However, they are limited in one or more ways. We have some of them that only detect changes at individual interaction levels and miss out on kind of the higher level structural unit level changes. There are coarse comparison metrics available, but with those you cannot pinpoint the regions of change that you can zoom into. Some methods specialize only in a single structural unit or resolution like TADs. And finally, most can only make pairwise comparisons between two data sets at a time. So we developed a method called TGIF based on multitask matrix factorization to attempt to address these limitations. Now, non-negative matrix factorization, or NMF, is the core component of our proposed method. Just as you can factor a number into two smaller numbers, you can factor a large matrix into two smaller matrices. Now, why might this be useful? The smaller factor matrices can be used to co-cluster the row entities to the column entities. So for instance, if you had the Netflix challenge data of user movie ratings, you can find a group of movies, perhaps by genre, and groups of users who prefer those groups of movies. Formally, the goal of NMF is to find two lower dimensional factors U and V that will approximate the input matrix X when they're multiplied together. Now, to take advantage of the fact that in many domains, you have known relationship between the row or the column entities, you may wish to incorporate that information into the factorization process. Again, going back to that Netflix example, if you have information about the Netflix user social network activity, the factorization process may benefit from knowing people connected in the social network with similar interests and preferences. So in this constrained variant of NMF, we introduced this graph regularization term to encourage the factor, in this case V, to be informed by or be smoothed to the graph topology that's captured by the graph Laplacian matrix L. Now we extended this to a multitask framework, TGIF, so we can naturally compare multiple contexts. In our proposed multitask NMF method at TGIF, each task corresponds to a biological condition or a cellular state and has its own input Hi-C matrix. And then for each task T, our goal is to find U and V factors that will approximate X while being smooth to the task-specific graph topology. And in addition, the factors from tasks that are closely related in the task tree or the task hierarchy will be constrained to be more similar to their shared parent factor here. And in order to learn these factors that optimize this objective, we use a hierarchical alternating least square optimization, or HALS, a type of block coordinate descent with a guarantee to converge to a local minimum. Now, once you have those output factors, we can compare them in the same lower dimensional space, since the first column or the latent feature from factor U of task A corresponds to that of task B. So we can use these factors to cluster the genomic regions and then find regions whose cluster assignment has changed across tasks. Now, first, we apply TGIF to simulated data sets with known ground truth clusters. And to do this in the most general setting, we created 3 different asymmetric matrices with 3 block diagonal structures representing the ground truth clusters. And the within cluster signal was drawn from normal distribution with a mean of 5, and the background signal was drawn from distribution with mean of 1. And we experimented with increasing levels of random noise with negative values capped at 0. So here the input matrices are visualized when the random noise variance level is 6. Now we benchmark TGIF to other variants of multitask NMF. The first kind that we benchmark to, called joint NMF, is just a simple NMF applied to a concatenation of the input matrices along the shared dimension, yielding a single factor V for the shared column or feature space. Multiview Nmf is another one. It's basically TGIF without graph regularization and with a single parent shared across all of the tasks. We also compare TGIF without any graph regularization using a 2-level task tree seen here. And finally, for the full form of TGIF, we used a simple neighborhood graph connecting regions that are basically within connecting entries within 2 indices of each other. So the adjacency matrix of these task-specific neighborhood graphs are visualized right here, sandwiched in between the factors in the input matrix. Now we measure the recovery of the ground truth clusters with Folks-Mallows index, or FMI. FMI is a geometric mean of precision and recall. So the true positives used for calculation refers to the pairs of data points, so pairs of rows or pairs of columns, in the same cluster, both in the ground truth and in the predicted clusterings from the NMF factors. And the false positives, FP, refer to the number of pairs in the same cluster in the ground truth only, and the opposite for false negatives. Now, for each increasing level of random noise, we ran each method with 100 times with different random initializations and took the average FMI across the 3 tasks per run, and then we plotted the mean. We also plotted the standard deviation, but the standard deviation is very small and thin, so it just kind of looks like a line here. Now, as we increase the level of random noise in the input data, we see that up to a relatively high level of noise, so 6 here, Multiview Nmf, which is the baby blue colored dotted line, as well as TGIF without graph regularization, so noted as tree here in the orange dotted line, they recover true sample or row space cluster better than the simpler joint NMF, and so that's the solid blue line. At very large noise levels where kind of the foreground and the background signals start merging, joint NMF outperforms them, possibly due to the availability of larger amount of data in the concatenated input matrix. And as expected, adding graph regularization boosts performance, especially at very high noise levels. In the feature column space clustering, similar pattern is shown. Interestingly enough, graph regularization of the row space factor U, so it's these guys here, helps with the true cluster recovery in the column space using factor V. So it's these clusters here. And as we see in the TGIF, this line catching up to the joint NMF at the very high levels of noise. Next, we apply TGF to real Hi-C data, which is what we care about. We started with real genome-wide Hi-C matrices from karyotypically normal hematopoietic stem and progenitor cells, or HSPC, and these are bone marrow cells with the potential to become any blood cells, any type of blood cells. And then we also have Hi-C data sets from 2 chronic myelogenous leukemia cell lines, K562 and KBM7, and then the 2 leukemia cell lines shared a parent in the task tree. Now here we visualize the sub-matrices of interactions only involving chromosome 9 and chromosome 22. Now each TGIF cluster is represented by differently colored horizontal bars. So for instance, this yellow bar here represents the TGIF cluster that is neatly subsuming all chromosome 9 regions in HSPC, and the blue bar, a cluster corresponding to regions under chromosome 22. Now from the earlier slide, you might recall that the Philadelphia translocation between chromosome 9 and chromosome 22 is a hallmark of chronic myelogenous leukemia. And it's captured as kind of this increased interchromosomal interaction pattern pronounced in the KBM7 Hi-C data shown here. Now, TGIF detects a single fused cluster among all regions from chromosome 9 and 22, represented by this blue bar in KBM7, kind of reflecting the large-scale translocation between those 2 chromosomes. We next apply TGIF to even higher resolution count matrices from mouse neural development data with Hi-C data from mouse embryonic stem cells, MESC, and neural progenitor cells, NPC, and cortical neurons, CN. So here we visualize the interactions around the 85 megabase region of chromosome 18 and the TGIF clusters of those regions represented in these colored bars again. And we see this fused cluster in the region around the 85 megabase of chromosome 18 and cortical neurons, highlighted by this pink horizontal bar here. So we see 2 clusters, a green cluster and a yellow cluster around this region, and MESC and NPC merged to a single yellow cluster in CN. And when we look at the RNA-seq levels normalized to TPM across samples here in a heat map, we observed differential gene expression in and around the fused clusters here, accompanied by a change in histone modification levels that is associated with active enhancer activity. So that's H through K 27AC. You see kind of how the peak patterns changes, especially in CN. So taking this all together in conclusion, we've developed a multitask factorization method called TGIF to detect changes in Hi-C matrices from multiple biological contexts. We find that TGIF effectively recovers ground truth clusters in simulated data and biologically salient changes across multiple structural scales in real Hi-C data. Now we plan to further refine our method so that first we can identify and summarize statistically significant changes across multiple conditions. And we want to measure kind of the association between the changes detected by TGIF and changes in regulatory signals known to affect 3D genome organization. So for instance, like histone modification levels or transcription factor binding. And then we can use those measures and metrics to not only scan for the best combination of hyperparameters and tree structures, but we also want to be able to systematically benchmark TGIF with existing methods for detecting structural changes in Hi-C data. So I'd like to thank my advisor, Sushmita, and the members of Team Roy. Although we are working apart remotely, they're basically keeping me sane and productive. And of course, we're able to do our research thanks to the funding from NIH and the computing infrastructure that's maintained by the Center of High-Throughput Computing here at University of Wisconsin. And I know I'll be happy to answer any questions, but if there are any questions, comments, and feedbacks that I didn't get to or you didn't get to share, especially for those on the streaming, please feel free to send a tweet to me and Sushmita. Those are our handles. Thanks for listening.
Thank you so much, Dan. Really nice talk. So I'll just make an announcement for the viewers while you're typing your question. So, you know, in the QA function, you can upweight certain questions. So if you see other questions that you like, you can upweight them and then they'll go on top of the list. But while you guys are thinking, let me ask you one question. So in terms of, so your model basically tries to learn shared structure across multiple related data set.
Mm-hmm.
Do you think, I guess, is there a danger if the structures are really different across contexts? And how would you handle that?
That's a great question. So in fact, when we looked at the benchmarking with the ground, when we have the data sets with known ground truth clusters, having a slightly more complex tree structure didn't really give a very noticeable boost against just like having a flat tree structure where you have one parent across multiple tasks. So our hypothesis currently that we want to test is, in fact, this might be more beneficial if you do have like a much stronger task heterogeneity, and that's somehow captured in the tree. And that's when you might actually see the benefit of using a tree versus just kind of sharing information uniformly across all the tasks. But that's something that, yeah, we're looking forward to test. But that's a great question.
Okay, cool. So you have two more questions. One from Max Liebrecht. Oh, sorry, the one from Irene Kaplow just got upweighted. So I'll start with that one. So, hey, what's going on? Okay, so we'll do Irene's first and then we'll go to Max.
Sounds
Did
good.
you try comparing TGIF to network diffusion methods?
Interesting. So when we, I'm going to assume when Irene says network diffusion methods, it's kind of about actually smoothing out the network, and we can treat the Hi-C matrix as a network between, you know, different genomic regions. So that's often used actually as a pre-processing step, especially for very noisy and very sparse Hi-C matrices. I am not aware of how to directly use network diffusion methods to compare across multiple contexts. There are network alignment methods that uses diffuse networks, denoise networks, that try to align it and then tries to find changes among the network structure. This is oftentimes used in dynamic gene regulatory network detection. But yeah, for network diffusion, I'm not sure exactly how I would do it. I will get in touch with you somehow to Irene and see if she has any methods in mind that we can further benchmark to. That would be cool.
Great. Yeah, it's easy to find Irene. I think you can look her up.
Cool.
All right. Question from Max, how does TGIF handle the dependence of Hi-C on distance and intra versus interchromosomal context? For example, do you see chromosome-specific clusters?
That's a great question. Thank you, Max. So when we're detecting, so depending on the task that you have in mind, we will basically use different resolution of the Hi-C data. So if we're trying to detect chromosome level rearrangements or kind of large structural variants, then when we use the entire genome-wide Hi-C matrix, including both the intrachromosomal and interchromosomal interactions. And in that case, it doesn't really, we don't do anything additional to handle it. It's just, you kind of see the block diagonal structures. Each block essentially is it corresponds to a single chromosome, but if there's chromosomal rearrangements, TGIF will pick that up, and we don't do anything to try to account for it or to deal with that. So if we're trying to find CAD level changes or compartmental level changes, that's when we kind of zoom into a single chromosome and use a much more high level, higher resolution Hi-C matrix. In that case, yes, you definitely see an even more pronounced, now that you've zoomed in, you see a very pronounced kind of diagonal streak, I want to say, that kind of represents what Max is referring to as kind of the dependence that nearby regions tend to interact a lot more with each other. We also don't have to do anything to handle that. In fact, we have a paper under review. The method is called Grinch, that does single task graph regularized NMF. So it's not doing multitask, we just kind of do it one per task. And we find that Grinch actually handles kind of this distance dependence well. And in fact, that's codified in our graph when we use the neighborhood graph. So we kind of, in fact, leverage that information, but we're not hurt by the kind of that streak pattern that we see. Cool. And I see.
Okay, and then that's just Irene sending you a pointer.
Thank you.
Okay, so thank you so much, everybody. We are a little bit behind on the schedule, so we won't take the full break. So you have 6 minutes until the next talk. And while you're kind of taking a break, maybe I'll quickly just say that for everybody that's presenting in the next today, make sure you are logged in as a panelist. That way you can share your screen and we can see you. So if you are presenting and you're not a panelist, please get in touch ASAP so I can add you. All right, so we'll be back in 6 minutes and then we'll get started at 10:40 with another oral presentation.
To be parameterized using independent exponential distributions with some rate. So there's some lambda for each of these. And since it's a non-clock tree, we have many of these parameters to learn. So just from a computational point of view, we have our likelihood, we have our tree and model priors, and our evidence. And marginalizing here is intractable for 2 reasons. So one, we're dealing with a composite space. So it's a space of discrete tree topologies as well as a continuous space over branch lengths. And we have this intractable summation over tree topologies as well as an integral over the branch lengths in order to marginalize to get rid of the hidden variables in this setup. So this is where the intractability comes from. This is what is hard in this setup. So just to recap before we talk about the contributions, there are several distinct challenges here, right? So there's the challenge of what I call inference or marginalization, right? So that has to deal with this intractable summation over tree topologies. It can often be done by sampling. And then for a topology, you also need to deal with this integral over branch lengths. So we need to marginalize over the discrete and continuous components of this composite space. And then we may also be interested in what I call learning or parameter optimization, model learning, model fitting. So what exactly is this Q matrix that governs this transition between characters? What are the parameters for branch lengths? So in this setup, the parameters are that Q matrix and then the lambdas that define the exponential. And so in learning, we want to find these parameterized that maximize the data likelihood. This is somewhat of a chicken and egg problem because the likelihood, the marginal likelihood involves this intractable inference problem of dealing with summations and integrals. So there are really 2 families of approximate inference algorithms that tackle this problem. And I find a useful way to think about them is in terms of local search. So Markov chain Monte Carlo or random walk Markov chain Monte Carlo is a member of a kind of local search algorithm where you start with a complete tree and then maybe we perturb something about the tree. We change a branch length, we change an edge, and we look at, we need to evaluate the likelihood using Felsenstein's pruning algorithm and look at this ratio of target over proposal and see if the area that we've moved to has higher probability than the area that we were, right? So we're doing this kind of diffusion on this composite space of phylogenetic trees and you know, this, you know, you can show that this converges to the distribution of interest, right? So it's important to note that local search, MCMC here, can be used for both inference and learning. So I can sample from a discrete space and sample from a continuous space using MCMC. And then MCMC can also be used in a way for optimization by doing this kind of random walk on the parameter space. So perturbing the values of the parameters until my Markov chain converges. Right? So there are a family of tools for this, right? So Mr. Bayes is kind of a famous random walk MCMC implementation. I should note that in Mr. Bayes, I believe the task is really inference, not so much learning. The Q matrix is just has a uniform probability across transitions. There are more sophisticated ways to deal with MCMC. For example, Hamiltonian Monte Carlo, or this recent work on probabilistic path. Hamiltonian Monte Carlo, where you kind of deal with curvature of parameter space and try to avoid getting stuck in local optima. Right? So this is MCMC, is a local search algorithm. The other approach you could take is sequential search, right? So in sequential search or sequential Monte Carlo, what you're doing is you're performing inference. So you're marginalizing, you get an unbiased estimate of the data likelihood, of the marginal likelihood. But you need to use some MCMC step or some EM step for learning. And so a sophisticated family of algorithms that came out called particle MCMC essentially uses sequential Monte Carlo to write down an unbiased estimate of the marginal likelihood. So there's one SMC step, and then there's one MCMC step on the actual parameters. So using SMC for inference and then MCMC for learning. So one example of this for Bayesian phylogenetics is something called combinatorial sequential Monte Carlo. That's what we'll build on. And then there are also these nice family of connections between variational inference and sequential Monte Carlo, where you use sequential Monte Carlo to essentially form an elbow. So you form a lower bound on the log likelihood, and then you optimize that. So it's a way of using sequential Monte Carlo to do variational inference and simultaneously do both inference and learning. So these are kind of difficult setups because you're dealing with this complex multimodal distribution on this composite space of phylogenetic trees. So quickly, in a nutshell, variational inference is a way of approximating an intractable distribution with a tractable distribution, and in doing so, trade integration for optimization. So the cost function VI, the elbow, essentially involves an integral or an expectation. So there's an element of marginalization there. And in doing so, you exploit this hidden symmetry, whereby minimizing this KL divergence between Q and P is the same thing as maximizing this lower bound called the elbow. So this work builds on combinatorial sequential Monte Carlo, and the novelty is in using combinatorial sequential Monte Carlo, an inference technique, to define a variational objective, and seeing how that's different from classical filtered variational objectives because we're dealing with this composite space. So combinatorial sequential Monte Carlo builds trees by sampling nodes and branch lengths. So we'll sample from a partially ordered set. We'll pick two nodes to coalesce with a given probability. We'll assign a probability or an importance weight to a partial state. And it's important to note that here, there's not a direct way of evaluating the likelihood. So Felsenstein's algorithm gives you a way of evaluating the likelihood of a complete phylogeny. What we have right here on this chain of ABCD is just A and B together. That's not a complete phylogeny, right? So there's a technical point which I'll come back to. And essentially, there's a resampling step where we focus the computation of areas corresponding to high probability mass. So we're going to iterate not over time points in this instance, but over rank events. And you can just see how we build this tree sequentially for a number of samples, right? So K sampled partial states are drawn from a proposal distribution at each rank event. I have a way of computing an estimate of this probability measure using the importance weights and a Kronecker delta. And the weights are really this ratio of the target over the target at the previous time point, divided by the proposal, this plus term, and then there's an overcounting correction, which you can read about in the Wang paper. Resampling states conditioned on previous states ensures that the variance of the importance weight scales linearly as opposed to exponentially across time points. So we're left with this unbiased estimate of the marginal likelihood. And the idea here is to approximate this posterior to do the model learning part by forming a lower bound to the data marginal likelihood, whereby maximizing the elbow is equivalent to minimizing this KL between the proposal, in this case, the recognition model or inference network, and the target, in this case, the generative model. So we're left with an unbiased estimate of the marginal likelihood. And just by Jensen's inequality, you can see that you get a lower bound on that by swapping the log and the expectation. So this is written in TensorFlow. And a few technical challenges here that I'll mention are that essentially because this is a latent variable model, you can think of it as a kind of autoencoder. And in doing some kind of autoencoding variational Bayes or reparameterization, there's a nice way to handle discrete continuous random variables, reparameterizing. But discrete random variables is a little trickier. So the two approaches you can take are to drop the discrete terms from the gradient estimate. So you end up with biased gradient estimates, or alternatively to redefine Gumbel softmax random variables. So when we sample two nodes to coalesce, I'm picking from a uniform random variable. So what I'm doing is I'm taking minus log of a uniform and shifting with Gumbel noise. So let's, another point that I should mention here is essentially in how we evaluate, I'll go back for a second, how we evaluate the likelihood of a partial state. So we know how we evaluate the likelihood on a complete phylogeny. Maybe I'll go back to the Felsenstein spooning algorithm quickly here. And that's done by passing messages from leaf node to root. And then finally, the last step in this formula is essentially to take the inner product of the message at the root node with eta, which is the stationary state of the Q matrix, right? So what I'm left with at each internal node is a distribution over characters, and including the root, I'm left with a distribution over characters. And I want to go from a distribution to a probability, and that's done by taking the inner product with eta, right? So when I sample partial states, I haven't yet assigned a probability to a partial state if what I'm left with is a distribution over, in this case, the parent of A and B, right? So the technical challenge here is an extension of the measure, the measure on complete phylogenies to a measure on partial states. There are more partial states than there are complete phylogenies, right? And so one way to do this is to treat all disjoint elements of the jump chain here as independent, and to essentially take the product of each of these elements with eta, the stationary distribution of the Markov chain, and then multiply everything together. So that extension is called the natural forest extension, and it has the advantage of passing information from all taxa into the local weight update, right? There are other ways you could assign an extension of this measure from complete phylogenies to partial states. That's a kind of an open question. So let's take a quick look at some initial results here. So this is on primate mitochondrial DNA. So essentially there are 12 taxa of different kinds of human, chimp, gorillas, and apes. And what we see here is the log likelihood estimate as the number of samples increases. So you see with 4 samples on the left, I get a lower log likelihood estimate with more stochastic gradient noise. And as I increase the number of samples, I converge to a higher log likelihood estimate with less stochastic gradient noise. And as a comparison, probabilistic path Hamiltonian Monte Carlo is run on this data. On the right, I probably should have put the names of the actual taxa here, but you partition, so the data is basically partitioned into clades where you have old world monkeys, new world monkeys, human, chimp, gorilla, and 2 more taxa. So once again, you get a distribution over phylogenies, and this is one that's sampled with probability according to the importance weight at the final step. So to recap, this is the first method that uses sequential Monte Carlo to define a variational objective on this composite space of non-clock phylogenetic trees. We see that we get suitable estimates of the log likelihood as the number of samples increases. This is a multi-sample objective, like an I-way, except with a resampling step. Some interesting extensions would be to look at beta coronavirus data, for example. So it may be an interesting direction for future work. Yeah, I guess I'll open it up for questions. I don't know how I'm doing on time here, but.
Thank you so much, Antonio, for a really clear talk. There's 1 minute for questions, and we have one question. So let's get to that from Khalil. Thanks for the interesting talk. In the model, are the branch length parameters learned as well?
Yes, the branch length parameters are learned as well. So it's a non-clock pilot, yeah.
All right, so for interest of time, we'll move on to the next speaker in this session. So that is Mohit Goya. You can go ahead and start sharing your screen. And the talk is going to be about joint integration and discrimination for automated single cell annotation.
Hi, Antonio, can you unshare your screen?
Yeah, sorry, let me pull my Zoom here.
So we actually decided to not do pre-recorded because live talks work a lot better. And so far it's been smooth. So thank you everybody for kind of being on time and being here, and also being flexible when we run late for 1 or 2 minutes. You can go ahead, Mohit.
So is my screen visible?
Yeah, we can see your screen and your mouse.
Okay, good. Hi everyone, my name is Mohit. Today I'm going to present our work, Genes, Joint Integration and Discrimination for Automated Single Cell Annotation. This is joint work with my collaborators, Guillermo, Ilan, Mikhail, and my advisor, Idoya. So I think most of us present over here can agree that a lot of single cell RNA-seq data is being generated, and many applications have already come up and new applications are coming in every day. But one of the first and foremost step in all of the pipelines that utilize this data is of cell characterization, which basically means identifying the cell type for each and every cell in the data. Traditionally, this is done using unsupervised clustering algorithms, and this is followed by use of expert knowledge to determine the cell type of each cluster. Now, it has been shown that annotations produced in this manner are very hard to reproduce and very sensitive to the kind of clustering method used. So if you take out a few cells from the data, or you change the data distribution slightly, the clusters might change, and so will your cell types. But at this point of time, many annotated data sets are present, which have been annotated by experts. And these data sets, if combined with supervised machine learning techniques, they present a natural framework to annotate more data sets, because this is a classic classification problem. So the question is supervised machine learning a better alternative to do this? So let's look at data-driven annotation of single cell data sets. So we have a source batch where the annotations are known, and the objective is to label a new target batch where the annotations are not known. Now, somebody might just try to use off-the-shelf classifiers because this is a classification task. Turns out there are a few problems with using them. First of all, depending on how the data was collected, how the library was prepared, the two data sets might have different kind of technical variabilities, which will lead to batch effects. So as you can see in the figure, the two data sets have completely different distribution. And for that, we need an external algorithm which can do batch correction, and then we can do joint analysis on these data sets. The second issue is, unlike standard classification tasks, each cell might not belong to a distinct cell type. As we know that cells can undergo differentiation and might transition from one state to the other, the cells can exist in intermediate stages. And so we get clusters like this forming a trajectory or something, and it gets difficult to classify cells at the boundary. So one way to handle this is to just say that we are not going to say anything about these cells, and basically do not assign any kind of label to these cells. So there already exist many tools which can do data-driven annotation, such as CRUT, Actin, scpred, SVMreject, and many more. And generally, there are three components to these methods. The first is a pre-processing step where we take the source batch and the target batch and project them onto a common space where the distributions are similar. Then the second step is to train a supervised classifier on the modified source batch. And the third step is inference, where we predict the cell types on the target batch. And while doing so, wherever we encounter predictions which are of low confidence, we just label them as unassigned. However, there are two issues that we find with these methods. First of all, if you remember, the ordering is like this. You first perform batch integration, then you perform classifier training, and then you do inference. Now, whenever a new target batch comes in, you will have to repeat the second process every time. And that is inefficient. What we would like is to have our classifier fixed and only do batch integration followed by cell type inference. That would save a lot of time. The second problem is with the rejection scheme. Whenever we reject cells, most of the existing methods use fixed thresholds, such as a threshold of 0.9. Whenever your confidence is below 0.9, you will label it as unassigned. But different datasets can have different complexities, and different cell types can also have different complexities, and therefore a fixed threshold is not optimal. So in light of these issues, we present genes, joint integration and neural discrimination, and our main contributions are as follows. So firstly, Gene uses a novel asymmetric GAN-based batch alignment method, where we do not need to retrain the classifier every time. Our second contribution is that we do not use fixed thresholds, but we estimate cell type specific threshold for every data set, and we utilize those in our rejection scheme. Third, we have Gene Plus, which is kind of an extension of Gene, and it's just like an additional step that you can perform after doing everything. And it is based on self-training on the predictions made on the target batch. And it kind of improves performance, as we'll show in our results. So let's look at the asymmetric batch alignment a little closely. So just to remind you, the idea is to fix the classifier and only do batch correction at test time. So we have a source batch, we have genes, and we know the cell types. Now we train a classifier, which is based on a neural network. Specifically, we have two sub-networks, an encoder and a classifier. The encoder takes in the gene expressions and predicts the low-dimensional latent code. Then the classifier looks at the latent code and can produce cell types. Now what we propose is to produce the latent codes for the target batch as well, and somehow align them to the latent codes over here, and never change the classifier. So as you can see on the right-hand side, this is what we have initially. We have the source batch and the target batch latent codes. And what we can see is that they do not overlap. Now, we assume that there is an underlying mapping in the latent space. And if we can uncover that mapping, we can just apply that mapping onto the target batch so that the distributions are aligned. And then we can use the aligned latent code plus the classifier to get the cell types for the target batch. So specifically, we use adversarial alignment. This is inspired from methods of domain adaptation in computer vision. We have our source batch latent codes. We have our target batch latent codes. But as we know that they might in general not have a similar distribution. So what we want to do is we want to align the latent code for the target batch, which is denoted by this, such that the latent code for the source batch and this aligned latent code, they have the same distribution. And to do this, we use a pair of generator and a discriminator. The generator takes in the latent code for the target batch and produces this aligned latent code. The objective of the discriminator is to look at the latent code and tell whether it is coming from the source batch, it's a source batch latent code, or it is the output of the generator. And the objective of the generator is to fool the discriminator into believing that its output is basically source latent code. And after we train them alternatively, this training is finished, we can use this generator to transform the latent code for the target batch and get this aligned latent code. And then we can just use our classifier to produce the cell types. Note that we do not change this encoder or the classifier during this step. So let's look at a data set and look at the performance of gene batch alignment. So we have pancreas data set. We are going to visualize the latent code distributions before and after alignment. So this is what we have before alignment. As we can see that the source clusters and the target clusters do not correspond to each other. But after we do alignment, every target cluster is mapped onto one of the source clusters. Note that this is done in a completely unsupervised manner. And we notice that this mapping is actually the right one. When we evaluate the performance of our classifier before alignment, we see that many of the cells from alpha and many of the cells from beta are rejected, basically labeled as unassigned, because the confidence in the prediction is not high enough. But after we do this alignment, the same classifier only rejects a small proportion of the cells, which basically means that this batch alignment does work. So we do a quantitative comparison with the existing methods. We have 3 data sets over here. Each of them have batch effects. We are going to use 3 metrics: the raw accuracy, the rejection rate, which is the percentage of cells that are rejected due to low confidence, and the effective accuracy, which is the accuracy on the remaining cells. So first, let's compare Gene and Gene Plus. So we see that Gene and Gene Plus performs similar in the terms of raw accuracy, and Gene Plus is slightly better. But if you look at the rejection rates are almost half. That means you're going to lose out on half the number of cells. So that is why Gene Plus is slightly a better alternative. Now we compare against Seurat label transfer, which is a part of the Seurat library. We see that on PBMC dataset, the performance is slightly better for Seurat, but we are close enough. On the other 2 datasets, we see that the performance of Gene Plus is significantly better. And that means our batch alignment is actually superior to what Seurat LT does. We also compare with 3 other methods: SVM reject, scpred, and actin. Note that these methods do not have a batch integration module. So we use Seurat integration and provide numbers for both with and without integration. So if we do not perform integration, we see that a large fraction of the cells end up getting rejected in all of the 3 methods. And in terms of raw accuracy, while the cells are not rejected, the performance is, while it is similar on PBMC dataset, the performance is not as good on pancreas datasets. If we use Seurat integration before running these 3 methods, we see that the rejection rates reduce a lot, but in some cases they do not, and the rejection rates are still very high. But even after integration, if we compare the raw accuracies, on pancreas datasets, the performance is still high for Gene Plus. Lastly, we also conduct differential expression analysis. The idea over here is to look at the mistakes made by our classifier and try to understand what happens over there. Is it some kind of noise that is being captured, or is it a genuine mistake? So we take PBMC dataset. We look at the 2 cell types, monocyte FCGR3A and monocyte CD14. As you can see that there is a lot of confusion at this intersection. So we try to conduct the differential expression analysis over here. So we specifically choose 2 groups. One is where the monocytes are correctly classified. Monocyte FCGR3A are correctly classified as monocyte FCGR3A. And the other group is where these monocyte FCGR3As are misclassified as monocyte CD40. Now, if everything was okay and it was a mistake made by our classifier, then ideally these 2 groups should not express differential genes. But that is not the case. As we can see that based on the gene expressions, we can separate these 2 groups out. So this purple group is the G1 and the orange group is G2. Moreover, when we look at 2 marker genes, FCGR3A and CD14, we see that these marker genes are differently expressed in these 2 groups. And this basically shows that this is not some noise that is being captured by a classifier, and it is essentially it can potentially be some dataset annotation noise, and that is our main conclusion. So to summarize, we have Gene Plus, which provides state-of-the-art performance on the task of dataset annotation. Secondly, we show that our rejection rates are consistent across experiments. Note that you can control the amount of rejection rate easily. Our implementation for gene batch alignment can be run on CPUs and GPUs. Even on CPUs, we are approximately 5 to 6 times faster than Seurat symmetric batch alignment, which is already inefficient because of being symmetric in nature. Fourth, our differential expression analysis reveals that there might be potential noise in the cell type annotations. Lastly, there's one thing that we do not discuss over here is what happens when a new cell type is introduced in the target patch. Now, if there is a new cell type, then you can not directly align these two distributions. And it is interesting to see what happens over there. And what we show that while gene misclassifies all those cells, our mapping learned by the GAN is actually still meaningful. And if you're interested, you can go look at our preprint, which is available on bioRxiv. So thank you, everyone. Our implementation is available on GitHub. Our preprint is available on bioRxiv. And if you have any questions, please feel free to ask.
Thank you so much, Marie. Right on time. We have actually a couple of minutes for questions. We can wait a little bit as people are thinking about it and typing, but I'm sure there's many people that work on single cell data, and alignment is a really big problem. So let's see, don't be shy, you can ask questions. So here comes a couple. First one from Youngjin Park. How do you handle doublets?
So can you explain what do you mean by doublets specifically?
So doublets is when it's actually a single cell is not a single cell. It's two cells that are merged, and you're seeing the signal from both of them. And you don't know what doublet. You don't know which cells are doublets.
Right. So I think there are a lot of corner cases like this, and while we cannot specifically change or while we do not specifically detect these doublets, but what we prefer is to look at the probability estimates that we get from our classifier, and we hope that the estimates would be poorer in these cases. Now, they might not be, and in this case, you would have to deal with misclassifications, I think.
Okay, thank you. Two more questions. One from Omid. Have you used VAE for latent variable estimation?
No, we do not use VAE. We use GAN, which is Generative Adversarial Network. So I think the main difference in VAE and GAN is that you do not explicitly model the data distribution in the latent space. So no, we do not use VAE.
And I guess the answer is that you haven't compared them either. You haven't compared them.
Oh, I see. So I don't know if there's a method which, so there is a method, I think you're referring to SCVI, which does latent variable estimation in the latent space, but they do not have a method for cell classification, but we can essentially use their model for batch correction and then use some other model for classification. And that is something that is useful. Thanks a lot.
Okay, question from Gallup. Can you use this method to align single cells from different technologies? For instance, single cell ATAC and single cell RNA-seq.
So is this question in the chat? Because I cannot see in the...
So this is the panelist QA.
Oh, okay. So that's a very good question. This is something that we recently tried. And one of the issues with single cell RNA and SC attack data is that their scales vary a lot. Now, ideally, like I've seen people doing batch alignment with two different kinds of data, which is scRNA and SC attack. But in our framework, we cannot directly use it because this encoder is expecting data at least at the same scale. Now, if you because I recently saw data and the scale varies like in 10 to the power 6 order of magnitudes. So that is one reason that you cannot directly use it. But we are trying to, but it would be separate project, but you can use it, I think. Yeah.
Okay, and then last question from Alan. Can you explain the architecture of the generator? Any sense of what sort of transformation is being inferred to handle the domain shift?
Yeah, that's again a good question. I think it's slightly involved. And for more details, I would suggest go take a look at our preprint. But what we use is, in abstract terms, is we model the latent space as sort of a nonlinear shift. So we model two quantities, which is a bias and scale. And we predict the bias and scale for every cell type in target batch that would lead to alignment. But again, like, you should go and look at the preprint because it's slightly involved, and that is very important for all of this to work. And good question.
Okay, since we have one more minute, I'll ask you the last question from Ethan. How sensitive was the GAN to hyperparameter changes?
Yeah, so I would agree that GANs are very unstable because of the nature of their objective function. But so we tried actually many techniques because this doesn't directly work. And at the end, like we tried a very simple thing, which is we penalize the generator with L2 penalty, and the penalty magnitude is very high. After doing that, this loss converges, and it basically converges in every data set that we tested on. I don't have a theoretical reason as to why penalizing the generator would make everything work, but that is one insight that we got into it. And maybe if you're working on something like this, you can try penalizing the generator, because the task of discriminator is very easy if there are batch effects, but for generator to learn the kind of noise that is there in the data set is kind of very hard. So to avoid overfitting, you might want to penalize the generator. Yeah.
Okay, thank you so much, Mohit. I'll hand it over to Suin, another organizer who will be moderating the next few talks.
Okay, thank you, everyone.
Okay. All right, hi. So the next talk will be Pinar Dimechi. Yeah. Hi, yeah, she's going to talk about a new method to align single-cell multi-omic data.
Do you see my pointer? I'm curious if I'm able to point at stuff.
Yeah, we see it.
Okay, thank you. All right, so yeah, I'm Pinar, and I'm a graduate student at Brown University, and I'll be talking about our work on a unsupervised alignment algorithm for single-cell multiomics data integration using Gromov-Wasserstein optimal transport. So with the availability of various sequencing technologies, we're now able to capture different properties of the genome at the single cell level, such as transcriptomic and epigenomic states. This is useful to get a holistic view of the single cell genome and perhaps study things like the mechanism of gene regulation or cellular heterogeneity from different genomic perspectives. But with very few exceptions, applying different technologies on the same single cell is difficult. And for some combinations of the sequencing assays that need to access the same part of the genome, it's currently not possible at all. In addition, one cannot sequentially apply these methods on the same single cell either, because a cell is destroyed after any of these sequencing procedures. So what we tend to do as a result is divide a population of cell of interest into different aliquots, and then apply a different sequencing assay on each of these. And then to perform a joint multiomic analysis, we would need to integrate these data sets. But this is a challenging problem. First of all, we don't have any information on one-to-one correspondences between cells from these different data sets, and we often don't have that for features either, because we measured different properties of the genome in different cells. So this process yields disparate data sets with no information on alignment a priori. As a result, we need unsupervised algorithms that will align them without relying on any correspondence information. Previously, one way people have addressed this challenge is by embedding data sets in a shared manifold. In this case, we assume that data sets share a manifold because we have measurements from cells that come from the same population. So they will likely share some genealogy and will probably have similar cell type composition as well. There are two unsupervised alignment algorithms currently that take this approach, MMDMA and Nmf. While both of them have demonstrated quality alignment results, they require the users to tune 3 and 4 hyperparameters respectively. Unfortunately, hyperparameter tuning can be difficult to do in an unsupervised setting, where when we have sequencing data from different aliquots and no validation data on alignments to use for the tuning. So in our work, we demonstrate an algorithm that's more convenient to use in a fully unsupervised setting, while still performing competitively with MMDMA and Nmf in terms of alignment quality. Instead of the embedding idea employed by these two algorithms, we approach this problem using optimal transport theory instead. Optimal transport generally aims to transform one probability distribution into another in a cost-efficient way by solving for a probabilistic coupling matrix. This matrix describes how the two distributions are related to one another. In our case, we work with data sets with discrete samples. So what we do is we define empirical probability distributions over these samples and work with these. Then the interpretation of the coupling matrix for our discrete case is the probability of correspondences between data points from each domain. And this gives us a transportation plan to align data points from the two domains. In the classic optimal transport formulation, the cost function for the optimization is defined over the data points themselves directly, comparing samples across domains. But this requires that the data from the both domains live in the same metric space. This assumption does not hold true in our application, since we work with different measures from different sequencing assays. So the feature space is different. So we use another formulation called Gromov-Wasserstein optimal transport, which allows us to work with data from different metric spaces. In this formulation, the cost function is defined over the intra-domain distances using Gromov-Wasserstein distance. And with this, we compare pairwise sample distances in the two domains instead of the samples themselves. And when solving for the correspondence matrix, we consider how to move a pair of points from one domain to the other. Using this formulation also helps us preserve some local geometry, since relative pairwise distances between the samples are preserved upon alignments. So we named our method SCOT as an abbreviation for single cell alignments with optimal transport, and here is how it works. Given two domains, we first compute intra-domain distances to be used in the Gromov-Wasserstein distance computation, like I just explained. We do this by constructing k-nearest neighbor graphs based on correlations between the samples, and then computing shortest distances between the samples on this graph. We choose to use KNN graphs because we observe that it also helps us preserve some local geometry during the alignment and yields better alignment results in comparison to something like pairwise Euclidean distances. Then, given these matrices and the empirical distributions, we solve the following optimization problem, where the goal is to find the coupling matrix gamma. And each entry in gamma tells us what the probability is of aligning a sample from domain X from the other samples in the domain Y. And we find these by comparing the pairwise distances. And we assign probabilities between these points that will minimize the pairwise distances among alignments. Intuitively, what this captures is how moving, for example, points xi in the x domain to yj and xk onto yl distorts the original geometry, and we want minimal distortion. I just realized that I'm actually missing a summation point over here, but think of like we're summing over all of the pairwise points possible. We are also using an entropic regularization for empirically better results. So what the entropic regularization does is it allows us to split the correspondence probabilities between a larger number of samples. And this regularization constant epsilon and the number of neighbors k in KNN are the hyperparameters that our algorithms use. But we've seen through our experiments that the algorithm is pretty robust to the choice of k. So we're mostly tweaking epsilon here. And given the correspondence probabilities, what we do in order to visualize alignment and also to benchmark later on is project the points from domain X onto domain Y by using barycentric projection. So barycentric projection does a weighted averaging of all points in Y for point Xi based on the correspondence probabilities between Xi and the other points. So we applied Scott on both simulated data and also real-world sequencing data. I will first show you alignment results qualitatively by visualization, and then we'll give quantitative benchmarking results later on. Here I'm showing you two data sets. The first one is a bifurcating tree resembling what we might observe in a cell differentiation situation. Each branch here represents a different cell type. This data set was originally generated by the authors of the MMDMA paper. And the second one is a synthetic RNA-seq count data set we generated using a simulation tool called Splatter. We also have 3 different cell types here. For each of these data sets, we have 2 domains, and they have been both created by projecting a lower dimensional embedding onto different dimensional spaces. So we have one-to-one correspondence between the cells in each domain. And this is true for both of the data sets. But we don't use these correspondence information for alignments. We only use them for benchmarking later on. Here's the visualization of the alignments performed by Scott. At the top, we have the data points colored by cell type, and at the bottom, we have them colored by domain. As you can see, upon alignment, the global structure is pretty similar between the domains, and the cell type clusters are also preserved. So next, we applied our method to real-world single-cell sequencing data sets. But recall that for simulation data sets, we had sample-wise correspondence information to use for benchmarking. To have something similar here for the sequencing data sets, we used data from co-assaying technologies. This means that we have 2 different types of measurements from the same single cells. Co-assaying technologies are limited, but there are a few available. The first data set we have is generated using SCGEM technology, and that profiles gene expression and DNA methylation simultaneously on the same single cell. And the second one we have is from SNARE-seq. It does the same, but with gene expression and chromatin accessibility. So we pre-processed both of these data sets according to the details in their original publication. And both of these data sets have a mixture of different cell types, but SCGEM also shows a continuous trajectory. And here are, again, visualization of the alignment results by Scott. At the top, similarly by cell type, and overall, the cell type clusters are preserved, and at the bottom, they're by domain. So far, I've shown you some qualitative results by visualization. But to quantitatively evaluate the alignments, we use a metric introduced by the MMDMA authors called FOX-TTM, short for fraction of samples closer than true match. It uses this ground truth information we have on correspondences to assess the quality of alignment. So here's a visual explanation of how it works. Let's say we have these 2 domains to align, and this is the alignment that we obtain from the alignment algorithm that we have. So let's consider the green sample here. Its true match would be the other green sample from the other domain, but after alignment, it's more closely located to 2 points out of all 7 samples. So its FOX-TTM would be 2/7. So we compute this over all of the samples in both domains and average it over both samples and domains to get one quantitative measure called average FOX-TTM, like one measure per alignment. And the lower values are better because ideally every point would be closely aligned with its true match, and it will be 0. So we want FOX-TTM measures as close to 0 as possible. So here is a plot of average FOX-TTM for each data set after alignment with Scott, MMDMA, and UnionCom, and the arrows point at the best alignment results. So Scott gives one of the best alignments across data sets, and for the first simulation, it's also pretty close to the UnionCom results. So far, the results that I've just presented you are based on alignments performed after hyperparameter tuning, just to show how well each method would perform in their best case scenario if it could perform hyperparameter tuning. But the realistic scenario is that a user may not have any validation data on correspondences to use for the tuning procedure. This is what we call a fully unsupervised setting. When we were looking into what we could do with this in this scenario, we realized that there is a relationship between Gromov-Wasserstein distance of the aligned domains and the alignment quality as measured by FOX-CTM. The alignment quality is here on the x-axis, and the Gromov-Wasserstein distance is on the y-axis. It's in log scale for better visualization. So as you can see, it's not a perfect measure, but given a data set for better alignments, we tend to get better Gromov-Wasserstein distance between the aligned domains. And this is not really surprising because it is a measure for this similarity. So as a result, we developed a procedure to guide hyperparameter tuning based on Gromov-Wasserstein distance to be used in fully unsupervised settings. On the other hand, MMDMA and UnionCom do not have similar procedures for when no validation data set is available. So in a fully unsupervised setting, realistically, one would perform alignment with them using their default parameters. So this is what we did. We also ran each method on the same data sets in the setting to demonstrate how they would compare to each other in a fully unsupervised scenario. So recall that this was the alignment quality graph from before. This is how it changes in the fully unsupervised setting. As you can see, the hyperparameter settings actually significantly affect alignment results for all of these algorithms. But with Scott's procedure to guide hyperparameter selection, we're still able to receive acceptable alignments, whereas with default parameters, some of the alignments can be really terrible for MMDMA and UnionCom. Lastly, we timed each method for a computational efficiency comparison. So UnionCom and MMDMA have their GPU versions implemented, and they're significantly faster than their CPU versions, especially for UnionCom. And we thought that people would be more likely to use their GPU implementations, so we used those. But for Scott, we actually only have a CPU implementation currently. So we compared these three. We basically sampled an increasing number of samples from our largest data set, which was the synthetic RNA-seq data set we generated using Splatter. And then we timed each algorithm. But this does not include hyperparameter tuning time. So UnionCom has some disadvantage here in terms of scalability, while MMDMA and Scott are pretty similar, with Scott showing some small advantage over MMDMA. To sum up, we present a new and promising unsupervised algorithm for single-cell multi-omic integration. And in the hyperparameter tuning scenario, it yields alignments that are comparable to the state-of-the-art unsupervised methods, MMDMA and UnionCom, but it's also easier to use in an fully unsupervised setting where we cannot do hyperparameter tuning because it has fewer hyperparameters and self-guide tuning them in fully unsupervised setting. And it's also computationally scalable compared to what's available out there. So we have our preprints on bioRxiv, and our code is also on GitHub. We have been working on creating a documentation site using GitHub pages. It's still under construction, but you can check it out for more details on documentation. I'd like to thank my collaborators for their hard work and mentorship in this project, and please feel free to ask me any questions.
I can see one question there. Yeah. So Mohit, do you wanna say?
Yeah. So I have one question. So you had a graph where you compared the performances of different methods when in the fully unsupervised case where you cannot tune the hyperparameters. So I suspect you can select hyperparameters for your case by using the proxy distance. But
From a Wasserstein distance?
yeah. But what did you do for the other methods for in the
Yeah, so we only use their default parameters because they don't provide any sort of way to select for hyperparameters when no validation data is available. So we ran them in their default hyperparameter settings. But I think it's like one of the most realistic scenarios because after getting sequencing data from different aliquots, you actually will not have any correspondence information.
Right, makes sense. Thank you.
So there are several questions in the Q&A section.
Yeah. I'm seeing that. Yeah. So just to understand the intuition, in your cost function, how does maximizing the entropy of the coupling matrix X on the, oh, wait a minute. So X as a regularizer?
Over here?
Yeah.
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 So it allows us to, without the entropic regularization, the algorithm will try to find, with entropic regularization, the probabilities are split between a higher number of points for each sample from one domain to the other. So without the entropic regularization, if the highest probability is on the wrong cell, you will get an alignment that's like completely off compared to what it's supposed to be. But with entropic regularization, there is more averaging, I guess. But it also actually allows for faster computation. And that was shown by Marco Couturi in a paper titled Sinkhorn Distances, Light Speed Computation of Optimal Transport Distances. So if you're interested in the advantages for computational speed when using the entropic regularization, there's also that.
Okay. So another question is about the value of K in KNN graph. How does it affect the alignment quality?
Yeah, so I actually was, can you see this? Did the screen change what I'm sharing? Okay, so we actually looked at that with different simulation scenarios. I'm just showing you an example. But so we have different, the colors represent the alignment quality. So we plotted that based on different epsilon values and number of neighbors, K. It changes by K, but epsilon affects a lot more. This is from an earlier experiment that we had, but K has some effect, but very minimal.
Okay, great. Okay, I think we should turn to the next talk. It's already 11:43.
Okay. Yep. I'll try to answer in the Q&A section. Thanks.
All right. Okay. The next presentation will be by Rosanne Rao on the transformer protein language models. They are unsupervised structured learners.
Hi, thanks everyone for coming to this talk. I'm Roshan Rao. I'm a PhD student at UC Berkeley. I'm happy to present our work on transformer protein language models, our unsupervised structural learners that was done as part of my work this summer at FAIR, along with Josh Meyer, Tom Serkiu, Sergey Ovchinnikov, and Alex Reeves. Can everyone see my screen and is it all going correctly? Awesome. So our goal is going to be to predict protein contacts from the protein sequence. And the traditional way of doing this is via something called direct coupling analysis, where we would obtain a multiple sequence alignment from a pairwise or from a sequence database and attempt to infer edges in the corresponding pairwise Markov random field, where each node in the Markov random field corresponds to a position in the sequence alignment. So this was proposed a couple years ago, and one of the standard ways of doing this inference is via pseudo likelihood maximization, where we try to infer weights of the edges in this Markov random field that maximize the probability of a residue at position I in a sequence, given the residues at all other positions in the sequence. And the learned pairwise weight matrix should then correspond to protein contacts. So there's been a lot of work more recently around training a neural network directly on a protein sequence database. So we want to ask the question is, can we actually infer protein contacts directly from this neural network without training that's not trained on protein structures? So to do this, we're going to specifically look at transformer protein language models. So transformers consist of multiple layers, and each layer has different attention heads. And each attention head learns a pairwise attention map for sharing information between positions in the sequence. So you can already see that there should be a correspondence between these attention heads, these pairwise attention heads, and the pairwise Markov random field that was used that's traditionally used to learn protein contacts. Additionally, we optimize this transformer using something called the mass language modeling loss, which is taken from natural language processing, but actually bears very strong similarities to the pseudo likelihood loss. So instead of maximizing the probability of a residue I given all of the other residues, we maximize the probability of masked residues given all unmasked residues. This is actually extremely structurally similar, and you can use the mass language modeling loss in a pairwise Markov random field and also learn protein contacts directly. So in order to extract contacts from these models, well, extracting contacts from a pairwise MRF is pretty straightforward because the high values in the learned pairwise weight matrix actually correspond directly to protein contacts. So the question is, how do we extract contacts from a transformer? Well, our hypothesis is that just as the pairwise Markov random field learns contacts directly in its pairwise component, the weights, that the transformer should also learn contacts directly in its pairwise component, the attentions. The problem is that the transformer has multiple layers and multiple heads per layer. In the transformer we spend a lot of time looking at, and in this work, there are 33 layers and 20 attention heads per layer for a total of 660 distinct attention maps, not all of which are going to correspond to contacts. So which of these attention heads actually correspond or will actually correspond to protein contacts? So I'm actually going to sidestep this question for a little bit and then just pretend that we already know which attention heads correspond to protein contacts and take a look at those attention heads. So this is the attention head from the first head in the last layer of a particular trained transformer. So this is actually just the raw attention map for that head. And we see that it already correlates fairly well with protein contacts. If we use the attentions from this head to do contact prediction, then it actually achieves a long-range precision at L of 55.2 on this particular sequence without any pre-processing or any training on protein sequences. If we apply a little bit of pre-processing to it via symmetrization and then APC, then we see that this jumps up from all the way up to 67.2. And throughout this whole process, the model is, again, not trained on any protein structures. If we then look at a couple more attention heads, my slide doesn't seem to advance. Okay, if we look at a couple more attention heads, then we see that actually multiple attention heads all seem to correspond to protein contacts. We also see that they seem to learn slightly different information. For example, if you look carefully at the third head in this sequence, we can see that this seems to be learning short or medium range contacts. So its long range precision at L is lower, but the short and medium range precision at L should actually be somewhat higher. So why don't we just combine this information by taking an average over these attention heads? Now, if we average over, well, one attention head is not really an average, it's just what I was showing in the previous slide. 5 attention heads is an average over exactly the 5 attention heads I was showing in the previous slide. And 10 attention heads is an average over an additional 5 attention heads that we think are predictive of protein contacts. And we see that, again, as we simply average over more and more attention heads, we see a significant increase in contact precision. Okay, but this kind of begs the question of how we're choosing which attention heads to average over and whether there's a more principled way of actually choosing these attention heads. And the way we do it is by introducing a tiny amount of supervision here. So we want to train this model via a sparse regression, where we're going to learn a single weight for each attention head in the transformer. And our hypothesis is that most attention heads are not strongly predictive of protein contacts. And so most of these weights that we learn should actually be 0. So this is our full learned contact probability for the entire contact map. We learned 1 weight per attention head for a total of 661 weights because we have an additional intercept term in the transformer that we spend most of this time looking at. Most of those weights are driven to 0, and the sum over this weighted average is passed into the logistic function to obtain contact probabilities. And then we can train this logistic regression on a small number of protein structures. And when I say small, I really mean a very small number of protein structures. Because the logistic regression has very few parameters and is strongly regularized and doesn't really need to learn that much information, we can actually just train this on a single protein structure. So this isn't the protein structure that it was trained on. It's trained on a different protein structure, but it's still able to have a precision at L of 82.4 on this particular sequence. If we train on a few more structures, we get slightly more robust weights and can increase this contact probability or this contact precision up to 88 or 89.6 as we train on 20 protein structures. And this seems to about be the cap of what this logistic regression model can do. And training on more than 20 protein structures does not significantly increase contact precision. So this is kind of the full pipeline of our contact precision. We have a transformer that is pre-trained on a sequence database to do masked language modeling. The weights of this model are then frozen and then passed, and then the attention maps are passed after symmetrization and APC to a logistic regression, which is fine-tuned on a small number of protein sequences. So looking at some quantitative results, first of all, let's talk about some baselines here. So the baseline that we're going to compare to is Gremlin, and Gremlin implements the pseudo-likelihood-based optimization that is standard for learning protein contacts from sequences. And we compared to Gremlin in 2 settings. In the first setting, we're looking at Gremlin trained on multiple sequence alignments generated from the ESM training set, which is the training set that our transformer used, and it's a subset of the Unref 50. The second setting is Gremlin trained on MSAs used in TR Rosetta. So TR Rosetta is a paper which achieves state-of-the-art results on Cas13, and it generates MSAs from Unref 100 and additionally augments them with metagenomic sequences. And so we think this represents approximately an upper bound in Gremlin's performance on these sequences. So we're going to evaluate Gremlin along with a number of different transformer models on 14,000 test structures after these transformer models are trained on 20 training structures. And so we're evaluating a number of different transformers here that are trained by different groups on different data sets with different parameters. But we see pretty similar trends overall. First of all of these transformers seem to learn contacts in their attention maps directly. Additionally, we see a clear relationship between model scale and the ability to learn contacts in the attention map. So if we look at the 6, 12, and 34 layer ESM1 models, these models have very similar hyperparameters, are trained on the same training data set, and are trained until there is a convergence and validation perplexity. And so we can see that precision at L, long-range precision at L, increases steadily as we increase the scale of the model. We can also look at this ESM1B model, and what this is actually slightly smaller than the ESM1 34-layer model. It has 33 layers and has 650 million parameters as opposed to 670 million parameters. However, it's been optimized in a sort of Roberta-like procedure to minimize validation perplexity by tuning a bunch of hyperparameters like learning rate, dropout, masking pattern, and long-range precision and layer norm. And so by tuning these hyperparameters, we can actually see that a smaller model is able to achieve a significant increase in contact precision. Looking at some of the simpler techniques for extracting contacts that I was showing earlier, simply averaging over the top heads is still an extremely reasonable way of extracting contacts from this model. So when I say top K heads here, the way we pick those heads is we train a logistic regression, and then we look at the heads that correspond to the top weight values of that regression. We throw away the actual weight values and then simply take an average over those heads. We see that simply taking an average over the top 10 heads is actually fairly competitive with Gremlin, and that even one head achieves reasonable long-range precision at L across this data set. We also, again, don't really need to train on many protein structures. Simply training on one protein structure and then evaluating on all 14,000 protein structures achieves performance that is quite comparable to Gremlin, while training on 10 or 20 structures outperforms Gremlin, at least on average. So if we go a little bit more into depth, those were all average results over a large test set. So let's take a dive down and look at the actual distribution across these 14,000 sequences. So what I'm showing here is the full distribution of performance of ESM1B versus Gremlin. And each point here is colored by the log of the number of sequences and the alignment used to train Gremlin. So what we can see is that, again, when Gremlin is trained on ESM data, ESM1B generally outperforms for predicting both short or all short, medium, and long-range contacts. When it's trained on TR Rosetta data, ESM1B outperforms for short and medium-range contacts, but for long-range contacts, the distribution is much more mixed. So ESM1B has a higher long-range precision at L for 55% of sequences, but that still leaves Gremlin outperforming ESM1B on 45% of sequences, which suggests that these are actually learning somewhat complementary pieces of information, and that these could be useful as a feature combination to a downstream supervised contact prediction pipeline. So this figure breaks down the relationship between MSA depth and performance a little bit more clearly. So again, when we look at Gremlin trained on ESM data, there's a clear ESM1B clearly outperforms at all MSA depths. What's particularly interesting, though, is if we look at Gremlin trained on TR Rosetta data for long-range contact precision, we see something that's actually very intuitive, which is that Gremlin and ESM perform extremely similarly when there are at least 1,000 sequences in the MSA, which is the regime where we know Gremlin tends to perform very well. But when there are fewer than 1,000 sequences in the MSA, ESM1B seems to perform significantly better than Gremlin. Another result, which I actually think is really exciting, is the relationship between validation perplexity and long-range contact precision. And what we're showing here is the training trajectories of this model as throughout pre-training history. So each point in this line represents a particular checkpoint of this model as it is training on UNRF 50 with mass language modeling. And we're comparing the validation perplexity on a random split holdout of UNRF 50 to the average long-range precision across our 14,000 protein test set. And we see a pretty clear, almost linear relationship between validation perplexity and contact precision. So this suggests that driving down validation perplexity actually should directly decrease or directly increase contact precision, which is a really exciting result for future scaling work. So as we get models that are larger, or have better hyperparameters or are trained on more data, we should see that they're able to drive down validation perplexity further and able to pick up on more and more protein contacts. Another small caveat is that the ESM1B line is not actually comparable to the ESM lines because the masking pattern for the language modeling training is different. And so these result in slightly different perplexity evaluations. Sorry, here we go. And so the last kind of result that I want to look at is if we want to generate new sequences, we can actually, we want to ask if we generate new sequences, do these sequences actually preserve protein contacts? So we can do this by masking out some fraction of the input sequence, say 20%, filling in that input sequence using the model's predictions of the amino acids, and then repeat that process using the newly generated sequence. And we can restart this process with some probability in order to prevent the model from wandering off too far away from the original input sequence. So this probably doesn't generate particularly excellent sequences because it doesn't have that many constraints on the generated sequence quality, but it generates a lot of sequences very quickly, which enables us to generate like a 10,000 protein pseudo MSA that we can then plug into Gremlin in order to predict protein contacts. So if we do this and do this across all of the 14,000 proteins that we test on, it's definitely significantly worse at extracting contacts than directly predicting with either with Gremlin or with ESM1B attention. So the long-range precision at L from generating pseudo MSAs and then passing those into Gremlin is only 14.5, which is still higher than, for example, some of the transformers were able to predict. However, it definitely succeeds quite well on some particular protein families. So for example, on this family through QHP, what we're showing on the left are the true protein contacts. In the middle are contacts that are generated or predicted via our logistic regression model. And on the right are contacts that are predicted by Gremlin when given this pseudo MSA. So kind of in summary, transformers represent contacts directly via a sparse subset of attention maps. Mass language modeling perplexity is an excellent proxy for contact precision. Better hyperparameters and bigger models will actually lead to better contact precision. And the best models are competitive with and even can outperform the existing bioinformatics pipeline. And all of our models and regressions are, or at least all of the ones that are trained on the ESM models, are available on GitHub now. So I want to say thanks to the FAIR team, Josh, Tom, and Alex, who worked with me directly, as well as to some others, Sergey, who also worked with me, and then Hustis Daupris. Thank you.
Okay, thank you so much. So just one question from Anushri Arora. is the masking done for just a single residue of the sequence, or is it done for multiple continuous residues of the sequence?
So I guess the masking at training time is done on 15% of residues at random.
Okay, and then a question from Li Shan: are the heads selected tend to be from higher layers?
Yeah. So let me show. So this is a distribution of the heads that are selected, actually split up by training a logistic regression specifically to look at very short range, medium, or long range protein contacts. And we see that there's really two modes where most of the heads are either in the final 4 layers or final maybe 5 or 6 layers as well as a number of layers in the middle, layers 14 through 20 actually seem to be quite predictive of protein contacts, which sort of leaves open what the initial layers and these intermediate layers are doing.
Okay. Does the increase in the number of heads help the performance?
I actually don't know that we have a result on that. There's another paper at iClear which looks at single layer attention heads that I also worked on with Sergey, and that paper does have a tendency to, that paper does show that there is a relationship between the number of attention heads and contact precision, but it's not obvious that those, but those are only on single layers of attention as opposed to large models.
Okay, all right. Okay, thank you so much. So we will have a lunch break for about 35 minutes, and then we will resume the spotlight presentation at 12:40. And then I'd like to remind you that we will have a poster session at the Gather Town website. And then I think Gerald just posted the website on chat. Yeah, you can find the link there. Okay, thank you so much, everyone. So see you at 12:40 for the spotlight.
Awesome. UCSF. Amrita, are you here, or is somebody who's supposed to be presenting your poster here? Okay, I don't see you anywhere, so we'll just quickly go on to the next person, and if you happen to show up later, just send me a message and then I can put you back in sequence. So first up then will be Lutfi from Paris Tech, talking to us about nonlinear post-selection inference for GWAS. Take it away, Lutfi.
Thank you, Gerald. Do you guys see my screen?
Yeah, it's good.
Perfect. So it's my pleasure today to present kernel Psi, which is a post-selection inference framework for nonlinear GWAS. The idea of kernel Psi came from what you observed, which is the complete dichotomy between SNP level and gene level. What happens is that on the gene level, we have increased interpretability and biological relevance. On the other hand, we have also on the SNP level, we have annotations which can be interesting and offer a breadth of informations. However, when we try to combine those two and we try to use those lead SNPs to derive gene level statistics, this leads to invalid inference. Basically, we are cheating by looking into the data twice. And this is where post-selection inference comes into play by correcting for the initial selection bias. We start by selecting a model which depends on the outcome in supervised fashion. After that, we move to inference, where we try to derive the distribution of statistic of interest. It can be anything that you want. For example, for post-selection inference, most of the literature focuses on linear model. And one prominent example, the thing that initiated the whole thing, is when we try to derive the post-selection distribution of a normal coefficient for a selected feature in a lasso. If you try to go to the nonlinear case, this is not possible to do it in a closed form. And this is why we turn to sampling in kernel Psi, where we sample replicas of the outcome to derive empirical distributions. We have shown how this post-selection inference framework leads to superior performance compared to non-selective benchmarks. Finally, I just want to say that kernel Psi is a powerful and scalable method for simultaneous slip-level selection and afterwards gene-level inference. So, broadly speaking, post-selection inference can be interesting to improve both interpretability and statistical power. And for the broad community, we provide kernel Psi as a tool available on GitHub on both CPU and GPU. So come visit my poster at booth 19. Thank you for your attention.
Great, thanks, Lutfi. Next up, we have Hossein Mohsen from Yale, who will talk to us about neural net interpretability using activation tuning and personalized products.
All right, so hello, everyone. I'm Hussein, and I'll be briefly describing two complementary approaches pertaining to neural network interpretability. The first of which is activation-based neural tree tuning, which is a Darwinian approach where the fittest neurons will survive training while others will be removed during the process. Our original hypothesis was that we can find a way to keep just enough neurons to maintain the predictive power of the network, but also we can remove ones that might collectively be injecting noise and rendering the network less interpretable. As for the criteria we use to remove or keep a neuron, it's based on what I'll describe as the training activity of a neuron measured through the magnitude of its gradient updates. It turns out we can proactively estimate this magnitude using target distributions that we designed to maximize the gradients for each separate activation function. On each tuning step, what we do is that we measure how close a neuron's inputs to the target distribution are. The higher this value is, the higher the gradient updates will be, and accordingly, the more likely the neuron is to survive the training process. Prediction and predictive performance are not the goal, but we measured and assessed the effect of ANT on predictions in 3 different tasks: MNIST, drug targets for leukemia, and diabetes hospital readmission. And we noticed that we can remove 70% of the first hidden layer neuron in a 2-layer fully connected network and still not jeopardize the predictive performance. And similar results were obtained when removing up to 50% of both hidden layers of the network. As for the second approach, which is the personalized weight products, or PWP, it's an approach that calculates feature attributions to predictions using a post hoc summation of the outgoing weight path in the trained network multiplied by a data subset that can range anywhere from a single data point to an entire data set, hence allowing for an efficient calculation of both global and data point specific attributions. To assess PWP, we first compared the features it prioritizes when acting on the complete trained network to those selected by the Garson's algorithm, another method that uses weight matrix multiplication. And we noticed that PWP selects a higher number of biomedically salient features in both the AML and the diabetes tasks. Lastly, we examined the personalized attributions on the MNIST dataset, whether we give the method input the entire MNIST dataset, each digit separately, or only a pair of images pertaining to the same digit. And we realized that PWP applied on the ANT specified networks select the key edge features of each of the digits being fed as input, but also of the specific images being received by the network. For more details, I'm more than happy to answer your questions, and please feel free to visit our poster during the session. Thank you.
Great, thanks, Hussein. Next up, we have Mukund Sundarshan from NYU. who will talk about insights into RNA splicing using interpretable ML techniques.
All right. Can you guys see my screen?
Yep, it's good.
Okay, great. All right, hey guys, I'm Mukund from NYU, and I'm going to be talking about novel insights into RNA splicing from interpretable machine learning. One of the most important processes that mRNA undergoes is splicing. During this process, introns are removed from the transcript and the remaining exons are joined. Splicing is dictated by the so-called splicing code, which maps sequences to splicing decisions. Exons and introns contain splicing motifs, which are short sequence patterns that play a key role in splicing decisions and are not well understood. In this illustration, for example, AGGA promotes splicing. So how would one typically approach this problem? Typically, a convolutional neural network is fit to the data. They're often amenable for motif discovery as they consist of short filters that slide along a sequence. These filters help learn a parametric function that maps a sequence to a particular outcome, in this case, a splicing decision. But hang on, there's a major issue with reading motifs off of convolutional filters. The set of convolutional filters learned is highly fragile. If you train your model several times using different bootstraps of the data, you're going to end up with different filters. In fact, we even saw in our experiments that several motifs don't always show up. Just shuffling the same data results in, in different filters. So which of these trained models should we use to discover motifs? Are motifs learned from these filters biologically meaningful if they're not reproducible? It's hard to tell. So what can we do about convolutional neural networks missing important motifs? Well, here's one way to think about the problem. Assume each filter is drawn from some latent embedding space of motifs. Every time a network is trained, filters are sampled randomly from this space. In any given training run, convolutional filters associated with a single motif may or may not appear in the model. In our experiment, we find that simply increasing the number of filters doesn't change its behavior. So how can we capture as many unique motifs as possible? Here's what we do. First, we bootstrap the training data set several times and then fit a convolutional model to each resample. Using the bootstrapped convolutional models, we generate an embedding vector for every filter in the model and group filters by similarity. Then a representative filter is chosen in each group by selecting the one that has the highest average similarity with other filters in its group. Now let's look at some empirical results using our methodology. Using data from a high-throughput slicing assay, we identified 6 important motifs. Our method was crucial in identifying these motifs, as individual models typically fail to identify 1 or 2 of these strong motifs. We were able to recover these missing motifs as our algorithm aggregates unique motifs across bootstraps. So in conclusion, we recommend practitioners don't attempt to interpret any single neural network model. Rather, to get reproducible results, bootstrap your data and refit models to each bootstrap. So thanks for your attention. Please check out our poster and abstract.
Great, thanks. Next up, we have Morteza Saber from the University of Montreal on identifying causal variants using predictors of microbial phenotype from genotype.
Hello, thank you. Thanks, Gerald. I'm a postdoctoral researcher from Montreal University, and my work is on evaluating the feature selection accuracy in machine learning-based bacterial genotype to phenotype mapping. Mapping the relationship between genomic content of bacterial pathogen and its phenotype is essential to precision medicine. However, this process is challenged by multiple confounding factors such as population stratification and genome-wide linkage disequilibrium. So far, there have been 2 main approaches to solve this problem. First has been the genome-wide association study, or GWAS, in which this confounding factors is explicitly adjusted and controlled for. And recently, we have machine learning models, which despite getting very good accuracy, do not explicitly adjust for this confounding factor. So our question was that whether this machine learning model are actually capable of identifying and adjusting for this confounding factor, or they are just prone to identify the spurious associations that might exist due to these confounding factors. So to evaluate this hypothesis, we first developed a simulator called BACGWASIM, which simulates bacterial population using a coalescent model and bacterial phenotype using an additive genetic model. And in the first step, by simulating bacterial population across a range of evolutionary scenarios, we first benchmarked different GWAS-based bacterial genotype to phenotype mapper, and we identified that the linear mixed models are the best GWAS tools for bacterial genotype to phenotype mapping. And we use this model as the reference for benchmarking different machine learning models. If in the beginning step, we started with different regression-based and tree-based machine learning models. And the result, and we found that across a range of sample size and evolutionary scenarios, while there is a big variation in the performance of different machine learning models, certain types of machine learning models are capable to identify and adjust for these confounding factors as good or even slightly better than that of the best performing GWAS approaches. And we also noticed that while there is a variation for identifying the low effect size versus high effect size causal markers across all these methods, the machine learning models are capable to identify the small effect size causal marker better than that of GWAS, better than the best performing GWAS approaches. So this work is under progress, and we are adding the different deep learning models as well. And for further information, please visit the poster number 21. Thank you.
Great, thanks. Next up, we have Johannes Linder from UW, who will be talking about inferring nonlinear feature attributions with scrambling neural nets.
Yeah, thank you. Let me know if you can't see my screen. And so yeah, I will quickly present a feature attribution method for biological sequences that is based on learning input perturbations. So the task here is to attribute a neural network prediction to its input features. So figure out which part of the input consists of the most salient features for the particular prediction. There are many choices of attribution methods for sequences. So in silico mutagenesis, deep lift, CHAP, etc. And depending on the particular sequence predicted task, sequence attribution can be quite challenging. There are two particular challenges I want to highlight here, which is saturation effects that can occur in the neural network.
I started to cut you off, but I think you have some serious echo. Do you have two devices?
There are many choices of attribution methods for. Is it better now? Or hello?
It sounds like somebody's playing YouTube in the background.
That is not me.
Anshul, do you know how to mute everybody except for Johannes? Oh, there we go. Yeah, sounds better.
Oh,
yeah,
great.
Go ahead.
All right. Hopefully you heard some of it. Oh, sorry. Now I can't see my. Sorry, can you see my screen still, or?
No, just reshare.
So, you need to. You can also just start from the beginning if you like.
Okay. Yes. Trying to attribute a neural network prediction to its input features. And there are many possible choices of attribution methods. There are two big potential challenges. You can have saturation effects that can occur inside of a neural network that can hide important regulatory motifs. And you can have feature independence assumptions that many feature attribution methods rely on that can break. So here we're looking at a particular example of a 5' UTR sequence, and we have a neural network predictor that is trained to predict translational efficiency given the 5' UTR sequence as input. Now in this particular example, we have an in-frame start codon marked in red on the left side in the sequence, and a stop codon marked in blue on the right side. Together, these two form a NAND-like type of repression because they create an upstream open reading frame. So this is a weak 5' UTR. And the neural network can predict this very well. However, by inserting multiple stop codons into the sequence, we create sort of a saturation effect where the effect of the stop codon is hidden from an in silico mutagenesis visualization method shown below. So to get around this, we take a different approach where we instead try to learn good sequence attributions by perturbations. So the idea is to train an autoencoder network that we here call the scrambler that is trained to reconstruct and find the smallest set of features inside of the sequence that preserve the original prediction. So in essence, we let the scrambler output a sequence PWM for each input sequence, after which we pull discrete samples from this PWM and feed it to the predictor model. And we then optimize the scrambler such that sequences pulled from this PWM reconstruct the original prediction of the unscrambled sequence. At the same time, we try to maximize the entropy of the sequence PWM, hoping that we're only going to find the most important feature set. And this approach builds on earlier feature selection methods, but whereas previous methods have used perturbations such as fading, blurring, or zeroing out unimportant dimensions, we either raise or lower the temperature. That's essentially what the scrambler predicts at each nucleotide position in this PWM. And that is, as we show in the poster, leads to much better performance for sequence predictive models. And so again, if we look at the 5' UTR sequence, after having trained the scrambler on data, it can easily detect both the start and the stop codons for this example. And if you want to hear more, we're located over there where the star is, and we're poster 10. Thank you.
Great, thanks. Next up, we have Jack Lanchinton from the University of Virginia, who will talk to us on transfer learning with neural motif transformers for predicting protein-protein interactions between SARS-CoV-2 and humans.
Can you hear me?
Yep, sounds good.
All right, cool. Yeah. So this is joint work with my lab mate Arshdeep and professors Clint Miller and Yanjun Chi. So a protein-protein interaction denotes a process where two proteins come in contact with each other to carry out specific biological functions. And these interactions between two human proteins are subject to attack by virus proteins, which is how the virus overtakes physiological functions. When a novel virus, such as the 2019 coronavirus, or also known as SARS-CoV-2, attacks the human body, the problem is we don't initially know which proteins it'll interact with. And this presents a few problems. The first being that if we assume there are around 20,000 human proteins and 26 virus proteins in the case of the coronavirus, this results in 500,000 potential interactions. And while mass spec techniques can find many of these interactions, it's impossible to directly test all potential interactions. So computational methods are necessary if we want to actually look at all possible interactions. And the second problem is that this task is specifically difficult for testing a novel virus since there's limited or no interaction data for that virus. So our training data consists of only human-human interactions and human-virus interactions from previous virus proteins. So while some of these might be similar, it probably doesn't transfer directly to the new virus protein. So in this paper, we introduce a new model that we call the motif transformer, designed specifically for in silico prediction between a novel virus and a human protein. So given a virus protein and a human protein, our model gives a prediction of how likely these are to interact. And to fix this problem of this training testing issue where we don't have any training data for the novel virus, we propose a transfer learning approach where the model learns general protein representations in order to better generalize for testing novel virus protein interactions. So we see on several data sets, including our own curated data set from SARS-CoV-2 proteins, that our approach, particularly using transfer learning, outperforms previous methods for novel virus interaction prediction. So I'll be presenting at poster 50 during the poster session. That's it.
Great, thanks. Next up, we have Sanjit Batra from Berkeley, who'll talk about learning putative causal gene regulatory programs.
Hi, can you see my screen?
Yep, looks good.
So today I'm going to be talking about in silico epigenome editing. This is joint work with Jeffrey Spence and Yun Song at Berkeley. So CRISPR-Cas9 systems can help cure monogenic disorders whenever the underlying mutation is known, for instance, in sickle cell anemia. However, if the disease is caused by gene expression reduction, but we don't know the underlying mutation, we might want to use epigenome editing. However, the question now is, where do we edit the epigenome? In order to answer this question, we train a model that predicts gene expression from epigenetic tracks. To build such a model, we use the publicly available ENCODE data that assays many different epigenetic markers across many different cell types. Inspired by the Deep Chrome paper in 2016, we have a similar setup where for each gene, we center a window at the TSS of that gene and consider many different histone modifications as the epigenetic markers, binned at 100 base pair resolution, and predict the gene expression level. In contrast to the Deep Chrome paper, which does binary classification of the gene expression value, we predict directly the gene expression value in a TPM. And furthermore, in the Deep Chrome paper, they build separate models for each cell type. However, since correlations invariant across cell types are less likely to be spurious, we treat each gene in each cell type as an independent data point and build a single model across all the different cell types. The data set that we have corresponds to these 12 cell types and these 6 histone marks. Once we train a model, we get a Pearson correlation of about 0.65 across the different cell types on held out chromosomes. Similar to in silico mutagenesis in sequence-based models, we can perform in silico epigenesis with the trained model, where we slide peaks of different epigenetic tracks to measure their impact on the gene expression level. In particular, here's a representative gene where the x-axis is the position in the input context and the y-axis is predicted gene expression. If we consider the dark blue line corresponding to the H3K27 acetylation, we see that as we slide an artificial peak along the input context, we see that the gene expression increases maximally when the peak is associated at the TSS. Once we are convinced that the model picks up reliable biology, we would like to understand how to propose epigenome edits with such a trained model. In order to do so, we set up an optimization problem similar to activation maximization in computer vision, inspired by the recent Lididi paper. And we asked the model to increase the gene expression of a representative gene. And here again, we see on the x-axis the position in the input context of a gene, and the y-axis the change in the epigenetic features when we ask the model to increase this gene's expression. And what the model does is places peaks of H3K27 acetylation and H3K4ME3 at the TSS in the dark blue and the pink line. It also places peaks of H3K36 trimethyl all along the gene body in the red track, and it removes peaks of H3K9ME3 in the brown track all along the gene body. We're now working with collaborators to verify whether these epigenome edits can be validated in vitro. If you'd like to chat more, please come see me at Poster 40. Thank you.
Thanks, Sanjit. Last up, we have Andre Duke from Utah State University. We'll talk about visualizing high-dimensional and high-frequency electrical biosignals.
Okay, can you see my screen?
Yep, looks good.
Okay, hello everybody. My name is Andres Duque, and the title of our work is Visualizing High-Dimensional and High-Frequency Electrical Biosignals. And basically the goal of our work is to recover a low-dimensional representation of high-dimensional dynamical systems and be able to visualize in 2 or 3 dimensions the behavior of possible latent factors that are driving the process. So here we have a little outline of the problem setup. Basically, we assume that there are latent variables confined in a low-dimensional manifold that are driving the high-dimensional process. And the way we tackle the problem is to split the data in several time windows. And what we want to do is, for each time window, to try to find a latent representation of the process over that time window. So our approach is based on a distance introduced in empirical intrinsic geometry by Talmon and Koifman, which is basically a Mahalanobis distance computed over histograms of data. And we mix it together with the Fate manifold learning algorithm, which is based in diffusion geometry, but has shown to be more stable to compute embeddings in 2 or 3 dimensions than diffusion maps, for example. And we also introduce some information geometry distances in the context of diffusion operators, which usually are computed in diffusion maps. We usually try to compute the Euclidean distance, Euclidean distances in diffusion operators, but here we try with another distances motivated by information geometry. So here are some results of our algorithm, and here we compute our approach for EEG signals, and we try to analyze sleep dynamics. So as you can see here, we are basically able to find a latent representation that not only allows us to visualize different sleep dynamics and sleep stages, but we also can see clear paths and trajectories that are happening over time, and you can analyze how the patient of the subject that from who you are measuring is changing across time in different sleep stages. Here we also try with EMG signals annotated by hand gestures, and here as well we are able to separate the different labels and visualize how the process is behaving. So, for example, here we see how a subject starts with the hand at rest, performs a radial deviation, and then goes back to the hand at rest. So yeah, basically that's our work. We are in poster 9 and hope to see you there. Thank you.
Great, so that's it for the Spotlight presentations. I posted a link to the gather.town platform in the chat window. You can find it on the mlcb.github.io page as well. I just want to point out one specific feature of the gather.town platform. And so if we just quickly go back to the gather.town site, if you get lost, because there's a lot of posters here, there's about 48 of them, you'll notice in the center of this room, there's a little poster board called poster list. And so if you hit X on this poster list, you'll basically get a map of the entire poster room and a list of all the titles on the right. So I've added short titles above each poster. You can find the full poster titles on the right. And so you can use this to find the posters that you want when you get in there. And that's about it for the Spotlight session. So hope to see you guys in the poster session